मेजॉरिटी आपण राहिलेल्यात आहोत त्यामुळे आपण जानेवारीत आला की
followers of the rule, they are all stuck behind.
So now who will be there? So Dr. Kane, how are we going to go ahead? It's 9:50.
Uday, I was just speaking to Raju Karnik and all. So second session, he says, leave it to us. We'll manage chairpersons karke. Because they want to have all the talks first and then the discussion. Aisa unka sabka Akshay Mehta hai, they have decided. So I said it's okay with us. And first session, we will have talk and discussion as soon as the talk is over, because each one is separate talk sort of a thing.
Correct.
And
looks good.
Yeah. So how I go? I'll just talk about this. Dr. Sadaran Shetty called me and he said, you talk about CSI, talk about the program, and then talk about the first few speakers, because they are our guest speakers.
Absolutely.
Yeah.
So just say about the CSI that this is in continuation of our meetings, and then you can introduce the subject and what are your thoughts on the subject. And what do you envision as the future of genetics? What do you think are the areas of which will evolve, are already established and will evolve in genetics? What do you think are the likely implications for cardiologists? Why we should learn this subject a little more in depth? So Karun, you can introduce and give an overview of this topic, particularly specific to this meeting.
Yeah, tell me one thing. Now, chat में कुछ questions वगैरह आता है तो वो कौन?
You have to take it. You are the program moderator. All questions you have to take it. Every single question you have to moderate.
Okay. So you will take over only at the final stages then?
Yeah, I will just propose the vote of thanks at the end.
Whatever.
For CSI members.
But already the chat is booming. Some 3 chats. I will see what it is. Advance Happy New Year and Christmas. Yeah, good. Yeah.
So we will start. Swapnil, are you there, Swapnil?
Sir, yes, I am there along with my colleague Rituraj.
Barabar. Swapnil and Rituraj. If anyone needs any help among the members, make sure I have given them your WhatsApp number. Don't ask them to call you, and they should not call Dr. Kani in between, because he'll be busy looking at the speakers and the moderations and the questions. So make sure you please take care of them. बरेच काय सिनियर कार्डियोलॉजिस्ट असतात त्यांना थोडी डिफिकल्टी येऊ शकते रजिस्टर करायला ना मुक्त.
नो प्रॉब्लेम सर, आय विल जस्ट सेंड माय कलीग ऋतुराज नंबर आल्सो.
ते करून टाक. एक सीएसआय मुंबई ब्रांच वर ऋतुराजचा नंबर पण व्हॉट्सअप नंबर टाक and make a mention to WhatsApp, not call.
Yes.
So that they don't bother you and then you are not on audio and all these things.
Done, sir.
Please put both the numbers as Uday says rightly, because it's difficult for people to understand. Your pehla registration, mag tula mail yeil.
Correct.
Because I also thought it is the same link, but then the link is different from the original.
And then what will happen is they'll go on calling Dr. Kani, and then he will not be able to focus on the meeting. So that should not happen.
Sure, sir. And if you get any call, just pass it.
I will. I'm there all the time.
I will do it. Yes.
Yeah, and make sure that we have everyone muted. You'll do it on your side.
Yes, done, sir.
Audio muting.
Yes.
Video कोणाचा चालू असेल तर आपल्याला काही ते बॉदरेशन नाही फार. आयडियली तर सगळ्यांनी म्यूट करायला, पण ऑडिओ 100% नको व्हायला, नाही तर इतका डिस्टर्बन्स होईल, वी वोंट बी एबल टू हिअर आउट प्रॉपर्ली.
सर्टनली.
Okay, so 2 minutes to go. At 9:59 you give the count. Register kar. The first talk is by a geneticist.
Hello, can you hear me?
Good morning, very good morning, welcome. Can you hear us?
Yeah, I can hear you. Can you hear me? I can see you.
Yes, I can hear you very well and clear, sir. Thank you.
Very good, thank you. But my video is not started, so I started it here.
No, your video, I can see you.
But anyway, I can't see anything here.
No, it should be, yeah. So are you muted on?
No, yes, now I can see myself. I can see Rajiv Gupta also.
Yes.
Good morning. Good morning, sir. Good morning.
Good morning, everyone.
Good morning, everyone. Yes.
Good morning, Dr. Gupta. Welcome to this program.
Such a great pleasure to be
looking
back
forward
among
to your talk. And
thank you. Thank you.
Spending some bit of genetics, which we always, as some seniors put it, that we always put it under the carpet whenever something is discussed about genetics. We move forward.
Correct. Same thing.
So
Blind eye. Turn a blind eye to complete genetics. Anytime anything comes on genetics, pass it off.
Let's see what we can make out of it now in the 21st century. So let's see.
So the thing is like this, means we have 30 minutes for you total.
Yeah.
So you can give enough time for discussion. So you decide how much you want to talk and how much you want to leave for discussion.
20-minute talk, 20 or 25 minutes, 20-minute talk.
5 to 10 minutes at least discussion.
Yeah.
That would be great. Yeah. Because a lot of people I expect will have basic queries, which may be simple and absolutely basic also, but I think we should answer them.
Yeah. All right. Yeah.
Good morning, sir. It's 10 already, so we can start.
Okay.
Yes.
So I'll share my screen.
Yes, sir.
Yeah. Okay. Am I visible and audible? Means my screen more.
Yes, sir. Yes, sir.
Okay. So tell me when to start.
You can start, sir. We are already live.
Okay. Good morning, everybody in Mumbai and all over India and maybe even abroad, because we are expecting people to watch us this program from all over multiple places. So welcome everybody, and you can join in as it is convenient to you. We are here to host you for this program for the next 3 hours. This program is unique because first time Mumbai CSI chapter, we are a very energetic group of people. I'll share with you the. So we are a very, very energetic group of people with Dr. Sadanand Shetty as president, Dr. Ajit Desai as president-elect, Dr. Uday Jadhav as the secretary, Dr. S.B. Gupta is immediate past president, Dr. Brian Pinto and myself are vice presidents, Dr. Rajiv Bhagwat as treasurer, and Dr. Anand Rao joint secretary. And importantly, we have a number of executive committee members. I won't go into the details. You can read the name, but they are so active and so energetic. And they are the young guns, and we are the young at heart guns. So we all form programs, and Mumbai CSI chapter is having a new outlook since Dr. Uday Jadhav and Sadanand Shetty and all of us have joined this committee. So we have regular programs which are attended by a very, very large number of audiences, and we try to make this all India level so that people can benefit from all nooks and corners of the country. So that is the whole idea. So today's program is unique because first time we are discussing something about genetics, as some people rightly put it out. Whenever genetics comes in the picture, genetics, statistics, most of us who are seniors who are not exposed to those things, we generally try to push over and go to the next step. But no, today we are going to learn this from some of the experts, and many of our people are also very good at statistics and genetics. So they are part of our panelists and the chairperson. So we are going to have a good fun. So all of you, please join us and put your questions in the chat box. And whenever the talk is going on, request that everybody should mute themselves. Otherwise, you'll be muted by the organizers. So this is the program in general. We have 2 sessions. The first session will be mainly the speakers will be geneticists who are practicing genetics day in, day out. And the second session will be by all our cardiologists from Mumbai who is who of cardiology in Mumbai. And our chairpersons are also very, very senior cardiologists, right from the first cardiologist of Mumbai city or maybe the country who DM cardiology, including lots of professors and head of departments and senior cardiologists with tremendous experience in the field of cardiology. I'm not going to name them because Mumbai CSI chapter being the host, most of our people know our people. So what I'm going to do in the next few slides is introduce our guest speakers so that our people and everybody knows about them. The first speaker is going to be Dr. Snehal Malakkamed. She is a consultant clinical geneticist with postgraduate training in pediatrics, and she has done fellowship in clinical genetics and practices genetics every day in Navi Mumbai at her clinic in Nerul and also at Apollo Hospital. And she's attached to many other hospitals. She has not put up that list. Since last few years, she is consulting patients in genetic specialty from all faculties of medicine. from premarital, preconceptional, prenatal, pediatric, adult. So this is all Greek to me, Snehal, but we want to understand from you when you give your talk. Invited as a speaker on topics related to clinical genetics at various clinical meetings, award posters and case presentations at national and international conferences, as well as publications. So we look forward to, because she's in Mumbai, she's the one whom we are going to fall back on when we learn about this and want to access genetic information or the genetic test. So she's our local person. Then we have some luminaries. We have Professor Shubha Farke, MD Pediatrics and DM in Medical Genetics. She's a professor and head of Department of Medical Genetics, Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. So she's a very senior person. Her areas of special interest are pre and postnatal dysmorphology, genetic diagnostics, psychosocial issue, and genetic counseling. And she's a teacher for DM in Medical Genetics program for last 25 years. So you can't have any better person than this to talk to about medical genetics. She runs a 2-week long yearly training program for clinicians, ICMR course in medical genetics and genetic counseling for last 18 years to create awareness about genetics among clinicians. Number of publications. She has a book, Genetic for Clinicians. Maybe I would love to have this book after this thing is over so that I can go through this. And many of you might be wanting it. She's the founding president of Society for Indian Academy of Medical Genetics. So a lot of awards, a lot of accolades. Madam, welcome to this meeting, and we are looking forward to your talk. Thanks for accepting our invitation and joining us to enlighten us about genetics of the cardiological cases that we see in our day-to-day practice. Last but not the least, we have Dr. Rajiv Gupta, MD, PhD, Chair, Preventive Cardiology and Internal Medicine, Eternal Heart Center, Mount Sinai, New York Affiliate, Jaipur. He's the Chair, Academic Research Development Unit, Rajasthan University of Health Sciences, Jaipur. He's an ex-board member of World Heart Federation, Geneva. Co-chair, Hypertension Group, ICMR, New Delhi, more than 1,000 publications, 650 original articles, 100 book chapters, my goodness, over 1 lakh citations, articles in all the major journals, and a guest speaker at all the major national and international conferences. So this is what speaker list we have for the first session. We are basically practicing as clinical geneticists, and to chair these sessions, we have our own senior cardiologist and young active cardiologist. It's a group of combination of the two. So the first session will be chaired by Dr. S.L. Soneji, who is our senior most cardiologist, DM cardiologist from the city of Mumbai, and maybe from India too. Dr. Sadanand Chetty is our president of Mumbai chapter. Dr. Prafulla Kerkar, everybody knows. He is the just retired as a professor and head of cardiology at KEM and is the emeritus professor at KEM Hospital. Dr. Milind Farke is a professor of cardiology at Sion Hospital. I'm also going to tell you the chairperson of the next session because we will move. Dr. Ajit Desai is our vice president and a very, very senior cardiologist. Dr. Rajiv Karnik, Dr. Akshay Mehta, and Dr. Nitin Burkule are some of the experts of all aspects of cardiology. They are the people to go to when you have a difficult case and it's not the run-of-the-mill case. These are the people who sort out the issues for us in Mumbai with either their echocardiography skills, EP skills, clinical skills, and whatnot. So we have a very, very good chairperson and expert panel sessions. So right now I would put it and hand it over to Dr. S.L. Soneji to invite the first speaker.
Good morning, everybody. I think Dr. Kane has already introduced the speaker, so let's have Dr. Malakim to address us, and let's not waste time on that. Thank you very much. Malakmir.
Yeah. Thank you, Kane sir, for your so humble introduction. I initially only thank Cardiology Society for giving me this opportunity, Dr. Shetty and Dr. Uday Zadav. And very thank you for a humble introduction and continuous encouragement to Kane sir and Dr. Somnath. So I'll start sharing my screen. I hope it is visible.
Yes, you're very visible and clearly audible. Please go ahead.
So one thought which came to mind when we started arranging this genetics in cardiology in my mind was because of Corona, especially pandemics, we have been pushed into an era where we are too much hearing about genomes and variations and mutations and why so much of a different response in different patients for a single infective agent. So it has pushed us rapidly through into genetics, and that is why maybe this is at the right time that we address very simple issues, because it has been so come to practical use that any patient can come to any clinicians. It's not about specialties or super specialties, but interpretation of just RT-PCR, whether this will detect this coronavirus variation, whether this will detect this Omicron virus. So it has come to such a basic level, and we should have this understanding. So that may be the basic concept of introduction of genetics, and especially how it is applicable in cardiology. Be it what we have to gather from evolution of our knowledge in medicine, in genetics, because it started off with because man has always been trying to understand origin of disease, origin of pathology and physiology. So understanding origin of disease is constant pursuit of human beings. Right from whatever medical era, pre-medical or previous era, to right now doing angioplasties, offering medicines for a particular disease in cardiology. So I'm going to restrict my talk in the context of cardiology, because genetics has very wide applications in many specialties. And through this pursuit, constant pursuit, the pursuit started maybe what we knew in our textbooks in the school days, which started off with Mendel. So we have been introduced in school to Mendel's experiments. We had a little bit of genetics in our undergraduation and post-graduation, but not so much. And suddenly now we have to address these complicated genetic issues. So we have lots of gaps to be filled up. What Mendel did was a perfect experiment of joining purple and white flowers and getting out of it is pod shapes, see how can the seed colors can be matched up, how flowers can, if they are mixed up, what will be the progeny, if shapes are mixed up, two shapes are mixed up, what will be the progeny, how the mutation. And thus the experiments are so basic that they still stand true. So what now our knowledge is based on is Mendelian inheritance of diseases and non-Mendelian inheritance, meaning some of the diseases, they strictly follow Mendelian inheritance, where Mendelian laws are perfectly followed up. And some of the diseases we are completely want to unravel mystery, like our diseases with, multifactorial. You have atherosclerosis, you have diabetes, you have chronic heart disease, where they don't follow perfect Mendelian inheritance. So there are a lot of things which are into genetics, and I may not be able to cover up all. So some of the concepts and some of the case applications I have taken into today's topic. What we have gone as a specialist or a medical students are human being, symptomatology. The symptomatology, if I can have a pointer. Yeah. So a human being and the symptoms, whether the symptoms, the patient tells us the symptom, we examine the patient, whether it is particularly located to a particular organ like heart. Then slowly medicine evolved to get tissue biopsies and understand where exactly the block is there, whether this chest pain is because of particularly in this which tissue, whether it is artery or whether it is muscle. Then slowly we started digging up whether which proteins are affected, whether there are any cell membrane channels, whether it is affected, and then how these proteins are made up, why they are defective in a particular patient. And then we started going up. Actually, the life evolved from genes to human beings, and we started unraveling our medicinal mystery down, it's up, down, and we are going now down in a diverse direction. So how it matters, how genetics in cardiology, if I may say so, how it matters at a molecular level is, let us say a cell. We have multiple organelles in the cell. So in a cardiac disease, we have cardiomyopathies. So now we know that a particular affected mitochondria produce dilated cardiomyopathy. So we have mitochondrial diseases producing cardiomyopathies. We have cytoskeleton affected in some of the aortopathies, like we have in Marfan syndrome, like we have some of the fibrillin genes. If they are affected and they are mutated, we have a defective cytoskeleton, and that is why the vascular, the wall of the vessels is defective, and how the cytoskeleton is defective. centrioles. We have centrioles in the cell which are defective in it is applicable in many cancers, in many fertility issues. Some of the lysosomal storage disorders like Fabry's disease, which particularly presents in cardiology. And there are multiple other lysosomal storage disorders, cell membrane disorders, which come into myopathies, like we have muscular dystrophies, myopathies, where cell membranes are defective. We have channelopathies, where channel in the cell membrane is defective. So now what, and then endoplasmic reticulum. The endoplasmic reticulum is important for stress-related response. It packages proteins, and those proteins, if they don't respond in a stress, they are defective, like we have life stress disorders. So that is a particular focus in multifactorial disorders now, that endoplasmic reticulum and ribosomal disorders, how they particularly respond to stress and how they package these proteins under stress. And if they are defective, then they cause stress-related disorders. So that is a new focus of maybe research. So this is how we are going now from cells. We had tissue biopsies, and we entered into cells exactly where the pathology is located, which enzymes are defective, which metabolites are defective, troponins or CPK or etc. And now how these organelles are formed, how these organelles, which proteins are formed, and now that is why how these genes are making these proteins, and how these mutations in the genes are affecting proteins. So that is how the particular, that is a stage where we are now. So to simplify a human genome, we have 46 chromosomes, we have 22 pairs of autosomes, 1 pair of sex chromosome, we have approximately 20,000 genes in nucleus, and we have a different genome in mitochondria. We have in the cell nucleus, where there are nuclear genes, which control most of the functions and proteins, and we have a different mitochondrial genome, which forms a part of mitochondrial diseases, which is exclusively derived from the mother. So this we should understand in some of the mitochondrial disorders. Some of the mitochondrial proteins are still made by nuclear genes, but some of the mitochondrial proteins are made by mitochondrial genes, which are exclusively derived from the mother, and they don't follow Mendelian inheritance. And normally we have cytosine, guanine, adenosine, thymine. There is a complementary base pair, C perfectly with G and A is T. It is so perfect made in the evolution that there is no chance that it should go wrong. And this base pair, one base, I mean, this is C is one base, G is one base, and there is a base pair which is called as BP. I have written down these terms because these are exactly made. If you see even any virology or any bacterial report on mutations or any patient gene mutation, you will see this and read these terms. So this is a base pair. We have 1,000 base pairs which make up 1 kilobase. We have 1,000 kilobases which make up 1 megabase. Like we have in computers kilobytes and megabytes of information. Similarly in DNA, we have kilobases, megabases. So there may be 2 base pair deletion. There may be 1 kilobase deletion. That means 1,000 base pairs have been affected. There may be 1 megabase deletion or duplication or whatever. So that is the extent of deletion or duplication. We have 2 sets of 23 chromosomes in each of the gametes. We have so many billion of base pairs. So we have a huge data which we have to, search for. We still do not know much about it. What we know that less than 10% encodes protein. So 90% of the genome, what it does, we are still not sure of. 50% of genome contains repetitive forms of DNA. What is its function, whether it controls, we do not yet not sure, and that is a vast area of research still open for a debate. Less than 2% is associated with exonic functions, which is practically important in our clinical medical field associated with disease. So we are still addressing this 2%. I mean, there's so much that we have to still address. So basically, we have in the nucleus chromosomes, which are usually visible when they condense during the cell division. Now, if I just make up an analogy, this is a DNA and these are histone proteins. So that is how DNA is wound around histone proteins, and this makes up the chromosomes. And if you see the figure on the right side, we have how this is unwound. I mean, if I take a chromosome and take a wire of DNA, and then when we reach a last stage, from this we take up a DNA. So chromosomes are made up of DNA, parts of DNA are genes, and genes make up mRNA, which make up proteins. Now in the chromosome, we have centromere, and the short arm is called as P arm, long arm is called as Q arm. This is especially important when you read reports of microarray in some of the patients. So P arm and Q arm, and this is the centromere. So this is how the DNA is organized into chromosomes, which is important because when we say a karyotype, we are studying chromosomes. When we say a microarray, we are looking at a DNA at a particular level, but not at a chromosomal level. And when we are studying exactly DNA or mutations, we have to extract the DNA from the blood of the patient or tissues. We have to make it straighten. We have to then go on to genes and study the sequences. So that is how sampling and patient sampling and the particular methodology of investigations will apply. So this is important to understand how the DNA is arranged into chromosomes. And these genes now, when we come at the genes level, we have, say, a particular segment of DNA genes. And in the genes, we all know that there are exons, introns. We have studied in second year MBBS that it is transcribed into RNA. RNA is then spliced out. I mean, if these introns are removed, red, and only blues, which are exons, they make up mRNA, and then it is translated into protein. So any defects, see, for above this also defects cause disease or difficulties, and below this also at any stage disease can be there. So if there is any problem in the transcription, which we call it as error, we may form a defective mRNA. Any problem in splicing, defective mRNA, defective proteins, defective protein folding. Even if defective mRNA or everything is perfect, if the protein is not folded properly, there may be different difficult functioning of the proteins. And at the right side, if you see this figure, I've just shown the base pair. So a single base pair, which call it as point mutation, meaning a single base is mutated. G is replaced or C is replaced by some other thing. It is a point mutation. So this is how mutations happen. This is how this assembly is very important for a particular functioning. And when we study genes or in any of the investigations, now everybody must be reading about terms like epigenetics, genomics. So if genetics is slowly being evolved into a term like genomics, you may not hear genetics much, but you will hear genomic variation, genomic medicine, genomic center. So genetics was usually a study of genetics, particularly limited to genes. I'll talk about genome later. But there are some changes. If this assembly is okay, I mean, perfectly normal, but proteins are somehow abnormal. And we don't understand why everything is normal here, but proteins are abnormal. So whether there are any other genes influencing protein folding, whether there is any other factor like acetylation or histidylation of proteins, which is defective, and why this protein is. So this is called as epigenetics. And this is being increasingly talked about in cardiovascular diseases, where you may not get a particular mutation, but there are epigenetic factors into play which are affecting the protein functions. So now genome, what is genome? So if we have a complete assembly of genes is genome. So that is what you will read in investigation, whole genome sequencing. You have to order in some patients whole genome sequencing, meaning we are addressing whole genome. It is very rare. I mean, we are not using it very regularly. Now we have exons, introns. Now only exons make up proteins. We said that to less than 2% of genome. Now all the exons of all the genes, total exons from the genome make up exome. So you must have read on the reports exome sequencing. So meaning we are only sequencing exons which are important in protein functioning for the known disorders. And we have into research or into any clinical trials or drug trials, proteomes, meaning all the proteins in the cells which are made up from these exons make a proteome. So now proteome is into research. So you will hear the genome, exome, proteome in more of the reports or research or trials. And if we say earlier or still now, we have these terms in use as a specialties of genetics, like people or the laboratory or investigation studying chromosomes is cytogenetics. When we study at a molecular level, that is DNA level, it is molecular genetics. When we are still doing enzyme studies, biochemistry, like troponin studies or CPK or lysosomal enzymes or vacuolar enzymes, that is biochemical genetics, the laboratories are different. Medical genetics concerns about all the things in medicine. I mean, it may not be particularly so for disease, but you're still addressing and finding out the disease cause or research. So it is particularly to all the faculties, it is a medical genetics. Clinical genetics is a part of genetics which is applied into clinical practice. So it is important in a day-to-day clinical practice. Pharmacogenetics is coming, especially in countries abroad. You know that the particular medicines cannot be prescribed to particular genotypes. So pharmacogenetics is increasingly coming into. Cancer genetics is a different and very vast area where only cancer genetics, and it is really, a different specialty evolving depending on the very wide applications of newly emerging therapies. Proteomics, I have said, and genetic counselors are dealing between clinicians, and we have in India many genetic counselors and board. Especially it is more evolved into West because we have so many unanswered questions from families, from communities, and there has to be a bridging gap between clinicians and the community to make them understand in simple language. So that is what is genetic counseling comes into picture. But when we talk of all these terms and we venture into a sea of genetics, for what matters as clinicians to us are whether where to begin in my particular office practice when patient and family sitting in front of me, whether it is genetic. Whether I should utter this word genetics in front of the family or patient or should not. So how can I be sure where I should tell the family or patient that it is genetic and you may pursue it, where I should think of and see whether I should make up a puzzle and understand myself whether I can put up this puzzle together and make up, as a high risk for genetics. So to begin as a gross things, these are the factors where you can immediately say that yes, something can be genetics. Like you have targeted family history, 3 generation pedigree, affected family members, you yourself can get an immediate history. You have multiple affected family members for the same disease. You have a very early age of onset, like you have premature coronary artery disease without any factors, and you're suspecting what is in play, or very severe early disease like congenital diseases, intrauterine diseases, very early disease in a child. In cardiology also, disease in the absence of other known factors, you are venturing into an unknown territory, idiopathic. So nowadays the genetics has given us a very different dimension, what we used to call earlier as unknown etiology or idiopathic. So now what science says is unless you rule out genetics, you don't call this as unknown or idiopathic. So when there is absence of other all known risk factors and still want to know why it has happened, you can have a genetic dimension to it. And when there is a club of malformations or intellectual disabilities along with cardiac disorder, then definitely there are some genetic factors playing a role where these genes are at a primitive level of forming many organs. And sex predilection, like some of the muscular dystrophies, cardiomyopathy, where it comes into like Duchenne muscular dystrophy. And mothers of these patients of Duchenne muscular dystrophy, they are high risk for cardiomyopathies because they carry that dystrophin gene mutation. So some of the X-linked diseases like Fabry disease, so they're more particularly in males. So that is how when some of such history comes into your table, then definitely you should start thinking of genetics. And then you have to make up this puzzle. I mean, you have to put these all factors together. So to say what is 3-degree pedigree, let us say if this circles are females, squares are males. So if this female has come for a consultation and one step down, that is her child, one step sidewise, that is sisters and brothers, and one step up is 1st degree. So any step down, side or up is 1st degree. If you go 2 steps like grandsons, grandchildren, if you go 2 steps sidewise or up, like grandfather, 2 and 2, this is 2nd degree. And if you go 1, 2 and 3 steps down or up, it is 3rd degree. So when you say 3rd degree consanguineous marriage, 4th degree consanguineous marriage, that is how after drawing a pedigree, you immediately know what is a relation. In India, we have 3rd degree consanguineous, not in India, all over the world, we have 3rd degree consanguineous marriage is very common. 4th degree consanguineous marriage is very common. 2nd degree consanguineous marriage in some communities of South India where uncle marries niece are common. So we have these consanguinities common where after this pedigree or family study, you will know. So this may be the factors from history you can gather whether they can be genetics. Then you have clinical information supporting your findings, early findings, severity, biochemistry, some of the biomarkers pointing toward genetic disease, radiology, like you have CTs, MRIs, your echocardiography, and some of the tissue findings like in tissue biopsies. Earlier, nowadays role of tissue biopsy is limited where you are not getting genetic answers, but still we are using it. So when we put all these findings together in our computer mind, and then if the puzzle fixes up, then we say that yes, this can be a genetic family or a patient, and I should start exploring genetic angles to this particular case. So in cardiology, there are a whole list of disorders, which I've just chosen few, that there are aortic vascular diseases like Marfan, Ehlers-Danlos, Loeys-Dietz syndrome, especially thoracic aortic problems. Everybody knows, I mean, you are better aware of this, that abdominal aorta, more of atherosclerosis, and thoracic, you start thinking of vasculopathies. And then congenital heart defects, definitely we think of genetic disorders, but some of these disorders of congenital heart defects also apply in adults. Same family members may be affected, may not be affected carriers, or mildly affected. You have inherited muscle diseases with cardiomyopathies, left ventricular non-compaction, arrhythmatic right ventricular dysplasia, etc. And you have rhythm disorders like channelopathies, all these syndromes, and sudden cardiac death, be it hypertrophic cardiomyopathy, be it a very small aspect maybe, but be it channelopathy. So, and of course, heritable lipid disorders, which I have not listed, or there are too many. So these are the areas where you can immediately think of a genetic disease, and you may not gather many factors. So this may point to immediately to a genetic disease. To start with, if we start thinking of what samples we can send, I'm stating this so because in our setup, especially, I have listed down the samples like when we take a blood and we have to do a karyotype, we have to take blood in heparin tube, sodium heparin tube. And for DNA studies, DNA extraction, we have to collect the blood in EDTA tube. We can take cultural fibroblasts from skins. In prenatal, our studies, we take fetal cells from amniotic fluid, coronary tissue, fetal blood, etc. Pre-implantation genetic diagnosis, I'll just touch upon later. Bone marrow, especially in cancers, or earlier we used to do bone marrow studies in Gaucher's and all these diseases, and tissue biopsies. So I have listed these samples because what happens especially in Indian scenario is, we have many investigations patient have to do from their pocket. Many investigations patients are not ready to immediately tell. A patient is serious or sudden death, and family, and doctors also not in a position to utter about genetic diseases. So now what we have started doing from last few years is, in a sudden death, or where a patient is, say, ill and a lot of expenses in ICU, but you still want to do some genetic studies, we are taking blood of the patient, we are extracting the DNA, and we are storing at genetic laboratory officially and commercially. They have a separate form to fill up, where you can store the DNA for a period of time, say 1 or 2 years. And if the family comes later, then you can address the issues that your DNA is stored, whether you want to take up these investigations. And DNA storage is very cheap, approximately 1,000 to 2,000 rupees. So such facilities for practical applications also are there available with laboratories. Sample pickup by any of the laboratories is very easy now. From anywhere in India, from any of the hospitals. Earlier, we used to think about what temperature, which container, nothing of the sort. You just give the sample to the laboratory and it is perfectly picked up and stored. You just have to fill up the test requisition form very correctly. So this is very simple now. You don't have to worry about sampling. Tissue biopsies, in case if you get some of the cases where heart biopsies, tissue biopsies, you have slides sometimes make up, you can extract DNA from those slides. Now, a very basic investigation still important in some of the cases where you just say that there is a Down syndrome, you know that it is a chromosomal disorder, 21 trisomy, and you order a karyotype. You have a Turner syndrome, you have a coarctation of aorta, you see a bicuspid valve, you suspect a Turner syndrome, you order a karyotype. You have Cry-Duchat syndrome, you have some of the disorders like Klinefelter. I mean, the phenotype, I mean, the clinical features are so clear that you just see that this is Down syndrome, you order a karyotype. So karyotype is meant where you suspect that there is a chromosomal disorder, you're sure of. So still it is done in syndromic cases where it is perfectly fitting into clinical picture. But we have gone far beyond. We are doing now very fine investigations. Fish is also very old investigation. What this technology is important because it is used in current DNA extraction, separation, and higher investigation. That is why I have included this. Fish, people, everybody knows about that. When it is a part of the chromosome, say we want to look for trisomy 21 in one sample, and karyotype takes 3 weeks or 1 month, but we have to immediately give answer to the patient because of some reason. Then there is a particular strand or particular sequence in 21 chromosome which is separated out. To this strand, there are fluorescent probes which are attached, and those are detected. So if there is 21 trisomy, you will three signals. So it is just done in 72 hours. So FISH is done in Williams syndrome, FISH is done in DiGeorge syndrome, FISH is done where you know which part of chromosome you're addressing. So you should be very particularly saying and ordering the test to the laboratory that I'm suspecting this deletion or syndrome or this chromosomal problem, and I'm doing FISH. So it is particularly done when deletions are, because in chromosomal analysis karyotypes, less than 5 MB, million base pairs, difficult to detect. So there you make up a probe for that deletion and order it. So it can reliably detect deletions as small as 50 to 200 kb of DNA in some of the syndromes. This is a new technology very widely used in chromosomal microdeletions, microduplications, cancers, microarray, and you will hear it on and off now. So that how it is done is, this is patient's DNA and this is a reference standard human DNA in the laboratory. So there is a plate to which this reference DNA shown in red is already hybridized, and you extract the DNA of patient from any of the samples you have. You hybridize the DNA, and because I have said that there is a particular very fixed pairing C2G A2A, there is nothing, nothing can change this. So this DNA particularly hybridizes to reference genome and you get pairing. So, and you detect where there are various spots generated because of fluorescence or color on the plate, which are picked up by laser camera, and you get a computer generated information. So you say if you have patient and control 2 copies, you get yellow. If there are 3 copies, extra copies in patient as compared to control, you get green. You have less copies in the patient as compared to control, you get red. So there are so many colors from which the laser camera makes up a graph, which a bioinformatics team, which is very important in any genetic laboratory, interprets in view of current evidence, current whatever proved disorders, and then they tell us which chromosome, which part is deleted, extra, or inverted, etc. So this is how microarrays performed, and these graphs are generated. And this is still deeper, like we are now into alphabets directly at the molecular level, ATGC. So there is a graph generated by sequencing. Sequencing meaning you are studying a sequence of alphabets, sequence of 4 alphabets, ATGC. And if there is any change, key is replaced by say G, it is particularly shown as a graph or a color, and these colors, and that is interpreted by the bioinformatics team and of course the clinician, and then whether this mutation fits into your patient disease. So this is what a sequencing means. And this gene sequencing have commercially there are many pros and cons of various sequencing methods, which I'm not going to discuss to make it, keep it simple. We have single gene sequencing. Say I know I'm going to sequence this gene in this particular patient. I'm ordering this gene. Single gene disorders where a particular gene is known to particular disease, like achondroplasia, many diseases, only one gene, spinal muscular atrophy, we know only SMA gene, so we are only addressing SMA gene. So single gene sequencing. Duchenne muscular dystrophy, we are addressing only dystrophin gene. There is no other gene associated with dystrophinopathy till now, no research for this. So single gene sequencing. Multiple gene panels. We have a Noonan syndrome. Now, Noonan syndrome is associated with 8 to 10 genes. The channelopathies. There are multiple genes in channelopathy. You're not able to point out which channelopathies because we are not doing electron microscopy studies. We are not particularly doing cell membrane study. It is difficult. It is only into research. So you have so many genes to address. So you are doing multiple gene panels. Whole exome sequencing, which I've told that all the exomes and whole genome sequences. So these are the tests which are commercially available. But when you get the data, I mean, sequencing data, you have 20,000 genes. In a gene also, you have a huge ATGC information, you're lost in the information. So how to interpret it? So if you have a NGS data, now it is called as next generation sequencing, because as it evolved right from Watson and Crick, there are various methods which evolved, which gave rise to today's sequencing platforms, that is next generation sequencing, that is NGS. And we have a huge data generated. But as a cardiologist, we are only interested in a chunk of information which will be relevant for our patients in our practice. So we have to derive a chunk from this huge information and apply it into our practice. And while applying it, this is very important that we realize genotype, phenotype information. Genotype, that is internally coded heritable information of an individual, which can also be used to refer to which particular version of gene is present at a specific location. Say we are using GPS to go and we want to find the one mango tree which is showing on the GPS. So that particular location where that mango tree is located is a locus. The mango tree will give mangoes, but the two trees may be different varieties of mangoes. So a gene at a particular locus can have two alleles, one from mother, one from father. So alleles are two copies of genes. So we should understand these terms because it is usually mentioned in the report. So alleles are two different copies of the gene, and there are many alleles of one gene. Many copies can be there in different individuals. We are addressing at particular locus on the chromosome. Now, phenotype is observed as structural, biochemical, physiological characteristics of an individual determined by genotype, meaning there is a particular genotype and it is particularly depending one phenotype of one disease. So clinical symptomatology or a clinical presentation of an individual is clinical phenotype. If you are seeing on 2D echo, say a muscle hypertrophy or cardiac hypertrophic cardiomyopathy, it is a structural radiological phenotype. If you're saying raised CPK, raised troponins, raised some biomarkers in some of the storage disorders, it is a biochemical phenotype of that individual. So now you will more hear these terms henceforth, that there has to be deep phenotyping with genotype. We have to completely fix this puzzle, whether this gene is particularly associated in my particular phenotype. And that is why genetics is now called as more of personalized medicine, that I have this clinical phenotype, this radiological picture of the patient, this biochemistry and this structural defects, and whether this gene is causing disease in my patient and whether it will apply to whole family. So let us see few cases. This is a child who presented at 12 years of age with hepatitis. He was not responding to conservative management for hepatitis. Autoimmune hepatitis was suspected, and he was at a stage, like many months, he was suffering for hepatitis, and at a stage where they were considering liver transplant, it came to consultation. And there was history of in early childhood of operation of TOF, tetralogy of Fallot. Now, when you get tetralogy of Fallot, and I have shown this face because consent of patients has been taken for photographs and display. So this patient has a particular phenotype which may not be apparent now on the photograph, but there is a very high bridge and there is a little hypotaylorism and prominent long face, which is usually seen in 22q11 deletion DJOS syndrome. This is a very typical face, may not be, I don't know whether it will be, can be drawn from this photograph, but it is typical case of 22q11 deletion syndrome. Now, why this is important to know? Because a patient who is operated for TOF, very good till 12 years of age, and now suffering for hepatitis, because 22q11 deletion syndromes are associated with autoimmune manifestations. They can come with autoimmune hepatitis, autoimmune arthritis, pseudo-hypoparathyroidism, hypocalcemia. So just we did a fish study which showed interstitial deletion of 22q11.2 region. So if you see, there are two signals in the patient and one red signal, meaning other red signal on the 22q11 is not there because it is deleted and you are seeing only one signal. So this is seen in these patients, and when this particular patient has to be followed up, and then we detected hypocalcemia, hypoparathyroidism in this patient. Patient was treated medically and completely fine. So no requirement of liver transplant for this patient. He was completely fine on just medical management in just few days. So this is how particular and perfect precise diagnosis helps in management of these patients. But we should understand some limitations of the of our testing, like only some of the patients will be detected by FISH. Some of the patients may have because that FISH probe is very particular for that mutation. Some of the patients may not be detected in FISH testing. We may have to go for microarray. We may have to go for sequencing if it is point mutation for DGeorge. So that that limitation has to be discussed with the family first because if this test comes negative, doesn't mean that it is not 22q11.2. You have to still address by some of the other investigations. This is another child who came, I think, at 6 months. There was a history of previous 3 spontaneous abortions in the family and dysmorphism, which is apparent, like there is hypertelorism, there are infraorbital creases and upturned nose and long philtrum. And so when you say that the child is having supravalvular aortic stenosis, every cardiologist knows that it is associated with Williams syndrome and there is a hypercalcemia detected. So immediately they are sent for they're sent rightly so for Williams syndrome, and FISH was done and this was detected. But why I've included this case particularly is when you say a Williams syndrome, there may be hypercalcemia, there may be developmental delay, behavioral disorders. You have to monitor this child for this. You have to monitor this child for hypertension, and you will do so. But what happens when it is only supravalvular aortic stenosis? We'll see another case. Before that, I'll just touch upon these terms which are very regularly used on reports and in genetics is SNP, CNV, benign and or versus pathogenic variation. So what is SNP? Single nucleotide polymorphism, meaning you have nowadays we don't use the term mutation. We use the term pathogenic variation. Every report mentions it as pathogenic variation because why it is so? We have single nucleotide polymorphisms, like structure of the nose is same in everybody, anatomy of the nose is same in everybody, physiology of the nose is same in everybody, tissues are same in everybody, but we have different nose shapes. Skin is same in everybody, but we have different skin colors. So if there is at any particular locus, this is A, G and T at that particular locus in that gene are different in these 3 individuals, but that doesn't give rise to disease. It is just a polymorphism, meaning different forms of genes, polymorphs, different forms at a single nucleotide position. So that is SNP, which is not mutation. Copy number variation, you will see these terms in sequencing reports and microarrays. What is that? If this is a chromosome and let us say a gene yellow B, so if there is a deletion of yellow, it is deletion. If there is duplication of yellow, duplication, partly duplication, and inversion, B and C inversion. So these are copy number variation at this particular locus that may not suggest disease. So if there is only variation and vary in copy numbers of the gene, which is detected on the genome sequencing or a microarray or any of the investigation, may not be always so associated with the disease. They may be seen in normal population also. And there is a variation which sometimes is benign, not producing the disease. So earlier we used to call it as benign mutation. It is now called as benign variation. And pathogenic variation, there is a mutation which is known to cause disease. It is pathogenic variation. So this is a case which I was talking about, that there is 3-generation pedigree. This is a child at 2 years which came to attention because of supravalvular aortic stenosis. Now everybody it is referred as soon as the cardiologist says that supravalvular aortic stenosis, maybe some murmur, the child is asymptomatic, detected because it was referred for 2D echo. And moment you see supravalvular aortic stenosis, you feel that can be Williams syndrome, it is referred. But so the mother now, because patients are reading now, so as soon as you write supravalvular aortic stenosis, mother reads that whether it is Williams syndrome or not, what will happen to my child, whether there will be developmental delay. And now mother asks us whether this is Williams syndrome. I have not taken because the consent was not given for photographs, but this child was not perfectly well for that 2 years of age, developmental milestones were normal. The child was not having dysmorphism, calcium was normal, but mother, we did a 2D echo, had a valvular stenosis, and there were mild stenosis, and sister, no medications needed, completely asymptomatic. And there was history in their mother of some valvular disease. Now, many of the times at above 60, 50 years, you see valvular calcification, some stenosis, and you know that, yes, there was some valvular disease and she expired at 60 plus years of age. But this pedigree itself shows that this is autosomal dominant. Dominant meaning it is present in every past, can be passed into generation. Variable manifestation. This child has got at 3 years of age. Mothers are symptomatic, and the grandmother at 60 plus years of age, some valvular.
Dr. Snehal, may I interrupt a little bit? Yeah,
yes,
can you, because we are a little overshooting the
yes,
time.
Yes, sir. I'll just run through.
Yeah,
so the autosomal dominant William ALN gene is present in the same region as Williams syndrome, and the investigation may be different. And this, I'll skip on this case of hypertrophic cardiomyopathy, where the child was detected as Noonan syndrome and father had hypertrophic cardiomyopathy because it is also autosomal dominant. And this is a prenatal application of a case where the child, the fetus was detected to have mitral atresia and all these cardiac anomalies on 2D echo, and we had a abnormality on microarray. Now the mother had first abortion, second child was terminated because of these abnormalities, and in view of microarray showing this abnormality, we could do prenatal diagnosis. So in pregnancies, the application is we can do Korean villus biopsy at 12 to 14 weeks, 16 weeks we can do amniocentesis here. And nowadays in Apollo hospitals and in some of the hospitals in Mumbai, we have pre-implantation genetic diagnosis. Like we make up an embryo by IVF, we test that embryo by biopsy for this particular mutation, and if some of the embryos are normal, we put it inside in the uterus. So that is, so a team of doctors are needed into perinatal genetics when it comes to cardiovascular diseases. This is a case where 50 years old male presented with dilated cardiomyopathy. So this report, this phenotype suggests dilated cardiomyopathy, and the mutation was seen in TNNN2. But this gene mutation, as the laboratory says, are associated with so many other diseases, from which we have to make out that this family is associated with familial dilated cardiomyopathy because of this clinical presentation, and whether we should apply this as in children and other family members. So now we have applications into cardiomyopathies. We get pathogenic variations in so many genes. We have channelopathies where we have to address genetic mutations, neuromuscular diseases like Duchenne's muscular dystrophy, myotonic dystrophy, Friedreich's ataxia. We have heritable lipid disorders, very high carrier prevalence in population, and so many disorders in heritable lipid disorders. So to just take one example, how to approach a hypertrophic cardiomyopathy, we have 4 goals. One is you all define HCM by your criteria. You identify the categories of HCM, whether it is secondary, acquired, like athletes, or hypertension, whether it is associated with syndromic, when it is genetic, and non-syndromic, whether it is associated with single genes. Then you have goal 3 as provide the evaluation strategy for a proband to have genetic investigation, which particular genetic investigation. And 4th goal is how we'll apply to asymptomatic and symptomatic relatives at risk. So these you all aware of, because these are normal recommendations by American College of American Heart Society and College of Cardiology Foundation, that you can divide hypertrophic cardiomyopathy depending on these possible etiologies, and then you think of genetics and how you apply it into families. There are particular recommendations and guidelines, 2020 AHA-SSC guidelines, which you are all aware of. And when you obtain family history, if you pathogenic variation is identified, test at-risk family members. If likely pathogenic, you have to do segregation studies in the family. If unknown significance, still you have to study the family. No pathogenic variation, cardiac evaluations depending on the at-risk factors. And these are the recommendations still for heterozygous or not heterozygous members. So now we have to evolve towards, in cardiology, as a comprehensive approach, whether how establishment of specialized clinical cardiovascular genetics, in particularly in India, where you have so much variation in genotypes, so much variation in knowledge, so much variation in socioeconomic status and understanding and facilities. New test which Dr. Rajiv Gupta will talk about, polygenic response test in coronary artery disease, and what are the applications and what currently its status is, and pharmacogenetic applications of this test, emerging tests which are already available in the commercial domain, how it will benefit in clinical trials or drug trials which are upcoming, which are immediate application into our practice. So the thing is, we have to discuss with the patient. Pre-test counseling, very, very important. Discussion of benefits, limitations, possible outcomes. If test comes negative, what next? If positive, what next? And what is the impact on current treatment management of overall the family? cardiology. So to take home, as clinicians, they should balance their opinion based on clinical judgment, utility of a particular genetic test, trends of particular genetic test versus risks and outcomes against the routine medical test background, importance in family members, and importance in further management. So I think at this stage, wide consensus is needed, particularly in adult cardiology, for application of genetics into direct practice. Thank you all. Thank you for your patient listening. Sorry if I have overshooted the timing.
Well, thank you so much, Dr. Snehal. That was an excellent overview of basics of genetics. I almost felt like I was back in my first MBBS classroom also, and it is nice that you put everything in the perspective. But from our clinician cardiologist point of view, your last few slides were very relevant because we are aware of the American Heart Association statement as late as 2019. Of course, there are clinical cases to happen. Firstly, one from your talk itself, you didn't mention saliva. Is it of any value? People talk about when there was, I've heard the geneticists talk about you can just take your saliva and can get the entire sequence.
No, sir, not now.
So
that
that's
is
good.
I've not
That's well answered. That's well answered. My very important question to you is, as a cardiologist, which panel for a patient of hypertrophic cardiomyopathy? I'm telling you because which panel and which company out there? Is there any standardization? For instance, I just ordered one for last week or something like that. It was more in the context that from him, I would go on to his family screening. And as I understand in a hypertrophic cardiomyopathy, for him per se, we cannot really even figure out on the gene basis that particular hypertrophic cardiomyopathy patient, at least our guidelines don't talk about that, he will be a candidate for ICD, for instance. That's what we are really looking at. But from family screening point of view, we pick the disease early and we can do something about it. That's all right. So now tell me very clearly, for a hypertrophic cardiomyopathy patient, do I just order a cardiomyopathy panel? Or I know that there are about 7 odd things out there. I'm not really an expert, though I've had some experience with hypertrophic cardiomyopathy genetics way back to 10, 12, 15 years ago. We collaborated with Osmania University and with Narsimhan from Hyderabad. So tell me, what should I order to keep his cost to the bare minimum? Or should I just say, go to such and such a lab, I know Dr. Snehal, and you get this panel done? And how does it affect his pocket?
Yeah.
In terms of what panel I order?
Yes, sir. So to address your saliva questions, earlier they used to take saliva because the cells were loose. We can say bar bodies. I won't say that it is completely out. It can be still in research in virology, bacteriology when you get infections, but not particularly it's our clinical practice. That is why I have mentioned samples very clearly. The disk samples we can use.
Okay.
Now your second question, hypertrophic cardiomyopathy. I run through slides, but fortunately we have case discussion, so we can discuss it in more. But in hypertrophic cardiomyopathy, when you rule out syndromic associations, if there is syndromic association, which I did not talk about, and in today's platform, there is, I don't think much of discussion can happen about drawbacks of single gene versus gene panels versus whole exome. But if it is syndromic, you can choose that syndrome panel. Like you have a Noonan syndrome.
I'm talking about our standard adult practice. We don't get syndromic association as much. I'm talking about run-of-the-mill, which are invariably VUSs, as what you would like.
So in syndromic gene panels also, if you have ruled out all these things, you have standard cardiomyopathy panels. Now in standard cardiomyopathy panels, that is why I was talking about genotype phenotype correlation. Like you have MYC7 gene. If you have, so what they have come up now, you have to particularly say this gene and what type of hypertrophic cardiomyopathy features in that 2D echo, and which we are not doing in our practice. So that genotype phenotype correlation is very important when you get VUS, like you have some mutation, but you are not able to correlate whether this is causing in the family. So I think it is a very long discussion, but you can choose cardiomyopathy panel in any of the patients. If there is a pathogenic variation that is definitely fitting into that phenotype of the patient, it is definitely a mutation causing the disease. And if it is uncertain, then you have to study family members. You may have to do further studies which are backed up by research to prove that this mutation is causing.
Dr. Snehal, I'll interrupt you. I just have a pointed question to you as to by doing a few panels, like for instance, we do understand from hypertrophic cardiomyopathy, there are 7 or 8 genes out there which account for 60 to 70% of the cases. And there is no syndromic association. Now, my patient came back to me asking that he has investigated, he's done Google, he's got at least 3 or 4 companies out there which have given him a huge list, even the syndromic association list. I just ordered a cardiomyopathy panel, right? So you tell us that whether costing wise, it makes a difference as to how much you go into, or it's one package. It does not matter how many you test. We understand there may be VUSs or whatever. You tell us cost wise, how does it affect? Because I am the guy who's asking him to get it done.
Yeah, so cost-wise, now if you ask me, the costs are very less for gene panels. Lab-wise, if you ask me, you have to choose a lab which is accredited, CAP and NABL, and which is using ACMG criteria. So that laboratory should mention in the report that this variation has been classified according to American Cardiology of Medical Genetics criteria. Only those reports are valid. Other laboratories, if they're not using ACMG criteria, the reports may not be valid. And the third is, you have to always, like I have mentioned in the last slide, that sometimes you may get patients. I'm not very expert in cardiology, but sometimes you're not sure. You have to do CT MRI of coronary artery, that you're not sure. So similarly in genetics, you have some gray areas where you're not sure whether this will give rise to immediate benefit to the family. So in that case, you have to see how multiple members are affected, anybody affected at very low age, and whether this particular hypertrophic cardiomyopathy or channelopathy, which may have high incidence of sudden cardiac death. And so that discussion should go on into the family. So that is a very peculiar, I'll say, to the family and understanding of the family, how much they want to expend. That is how angiography.
We should decide.
If I can intervene, I feel if you want to order and you want to save money, it's best to discuss with a clinical geneticist like Dr. Snehal. She will probably help you before you order to save the money. Because they are there now.
I just want a pointed answer. If I order a small panel versus I order a large panel, how much of a difference it makes? You can say it costs 3 times more.
Not much.
Or 1 time more. That's what I'm asking.
So pointed, I will say not much difference. When it comes to less evidence in science,
I'm asking you a practical question. That's
I will just clarify.
Who? Milind wants to clarify something. Milind, go ahead.
So I was asking you is suppose we get so I'll just clarify this question further. What sir is trying to ask you is we have a patient in the clinic of a run-of-the-mill hypertrophic cardiomyopathy, 50-year-old, no syndromic associations, nothing. So as a starting point for screening for genetic panel, should we order the routine mutations or should we go into more depth and order the more advanced panels? Or as a screening, is it just enough to do the clearly identified pathogenic mutations? Because there are the cost issues.
No, this question was a little valid 1 or 2 years back, but now what laboratories have done, they have merged all platforms. So if you are ordering a gene panel, they are still looking at rare mutations. And if you are doing exome, so there are only 2 tests now. If you have to order gene panel or exome. So everybody is ordering whole exome and the platforms are almost same. So same data can be derived. So earlier we had this question, single gene, all these genes, but laboratories are merging platforms now. So you have to really discuss the case with the laboratory. And I think soon, maybe it may be very soon that all that platforms will be merged into same test. So till that time, I think there is a time to discuss test.
I mean, from what we have read, there is a lot of overlap between the genes of so many cardiomyopathies. So you may have hypertrophic cardiomyopathy in one family member and a sibling or some child may have a dilated cardiomyopathy. Because some of the genes are common. So that is something again which can be.
That is why I shown that case. I think Dr. Milind, we may be having the similar case. That is why I shown that case of TNNN2 gene. You have 6 phenotypes associated with that gene. Now you have to tell in the family that left ventricular, that compaction disorders where the child presents with congenital heart disease and the adult in the family presents with you that left ventricular compaction. So same disease manifesting in the child as a congenital heart disease, same disease manifesting as a sudden disease in the same family, and same disease as compaction. So it happens in some of the diseases. So that is why this history taking, and I took this as a puzzle that you have to really join this puzzle before we make up a clinical picture and order the test. I think we can address some of the issues while we discuss cases because it will be a discussion.
Milind and Praful, we have full interesting cases which brings out all these things. So please be with us. So let's move ahead so that and keep the major discussion at the end, because all these three experts are going to be there till the end with us. So otherwise, we will encroach upon the time for the other speakers. So can you invite the next person, sir? Yeah, so we will be now having Professor Shubha Phadke giving us a lecture, and I think it's a pre-recorded lecture, so we can listen to that. Welcome, Dr. Sadanand Shetty.
Hi. Yeah, you
Hello. Namaskar.
Yeah. Okay.
At the beginning, I shall like to thank Dr. Snehal and organizers for this opportunity to talk to the cardiologist. Because the audience is cardiologist, I shall give some relevant information and some illustrative cases from the cardiologist perspective. Since 1989, when I joined medical genetics, in last more than 3 decades, lot of changes have occurred in my personal life and in the genetics as well as in India. The most important relevant thing which I would like to mention here is completion of Human Genome Project, which was launched in 1989 and declared complete in 2003. Because of these developments in the Human Genome Project, lot of changes occurred in the clinical management of patients with genetic disorders. Human Genome Project, which completed with the amount of $3 billion for 3 billion base pairs, was compared to the biology's answer to the Apollo space program. However, after completion of Apollo space program, nobody is going to the moon again and again, not even one person per year. But after completion of Human Genome Project, practically now so many individuals are getting their genome sequenced, and that too at such a low cost so that it can be applied in the clinical practice. A time is not far away when every baby as soon as is born will have to get this first taste of sequencing of the genome, and which will be analyzed as and when required when the child gets some illness or for pre-symptomatic diagnosis. Human diseases, though we consider hardcore monogenic disorders and chromosomal disorders as genetic diseases, practically every disease of human being has some genetic component in its etiology in variable amount. And these include even susceptibility to tuberculosis or resistant to HIV infection. But mostly when we talk about genetic disorders, we talk about these monogenic disorders or single gene disorders where a mutation in a single gene will give rise to manifestations in that particular individual. They may be X-linked or autosomal depending on the location of the gene. Here you can see a child and mother with Holt-Oram syndrome. Their severity of upper limb involvement is different, indicating that even so-called monogenic disorders have some effect of other genes or maybe even environmental factors. The typical prototype of monogenic disorders is sickle cell disease, and it was described in 1910. The causative DNA variation was identified in 1957 in beta globin gene of hemoglobin, and after so many years, successful gene therapy has become possible in 2015, and also for gene therapy has been approved for beta thalassemia in 2019. So these changes really have made a marked revolution in the for patients and families with monogenic disorders. All of you who see Facebook must have got to know about gene therapy for spinal muscular atrophy, which cost about 16 crore rupees. And of course, the results are miraculous. The children who would die by 6 months to 1 year can now walk and sit with this human gene one-time gene therapy. Also, there are many other drugs based on the genetic modification for spinal muscular atrophy. However, the cost being very big or exorbitant, really we still advocate carrier screening of all families who plan for a baby, if they come in early pregnancy or if they come before planning pregnancy. Many genetic disorders like this, though we see often, however, they are very, very rare, and probably 1 per 1 lakh or 1 per 40,000, 1 per 2 lakhs, and some of the disorders have been described to only 10 to 20 individuals. So these rare disorders are very, very important, because for a particular family, the outcome is very bad, because many of them do not have a treatment. 80% of rare disorders are monogenic, and in 21st century, the government of India has taken and shown a great will to help these patients and families with the monogenic disorders. And Rare Disease Policy of India has launched in 2021. At present, it is a very small step, but it is in the right direction and will be helpful to the patients. For monogenic disorders, the rare disorders, the clinicians are also very rare. And still there are so many genetic centers in India. And this is because my teacher and mentor, late Professor S.S. Agrawal, had the vision to start medical genetics department in 1989 in Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. I joined there as a first DM student, and now there are more than 46 medical geneticists DM trained from our institute, and they are heading many medical genetics departments all over the country. Other institutes also recently started DM and DNB programs in medical genetics. Because the clinicians with training in medical genetics are few, and it is very important that every clinician should be empowered with the knowledge of basic genetics and molecular vocabulary, so that the results of these human gene project and various diagnostic and treatment facilities which have become available to reach the Indian patients. Everybody needs to have a good knowledge of genetics, and we are training clinicians at various stages of their career in short programs like a 15 days program, and more than 500 clinicians have come to our department to get us short-term training. As I said, genetic disorders mainly are labeled to Down syndrome and other chromosomal disorders, as well as monogenic disorders. So when I joined DM in medical genetics, my only goal was to establish prenatal diagnosis for Down syndrome and beta thalassemia. This is because these are a prototype, and once you establish investigations for one monogenic disorders, doing it for other disorders is easy thing. Thalassemia being commonest genetic disorder in Asian countries, many centers have started establishment of medical genetics from beta thalassemia like us, and because every patient with thalassemia, the clinical and diagnosis as well as hemoglobin electrophoresis based diagnosis is easy, but every family, the mutation or the DNA change can be different, and hence identification of mutation is very, very important before you go for further plan of prenatal diagnosis. The same thing holds true for other disorders like Gaucher disease, achondroplasia, where there are other diagnostic tests. But molecular diagnosis is very important for providing prenatal diagnosis. On the other hand, there are many disorders like Fragile leg syndrome, Duchenne muscular dystrophy, etc. There is no specific diagnostic test other than molecular diagnosis, and hence the molecular diagnosis has become a very important part of diagnosis of genetic disorders, which can present to any specialty and can involve any organ of the body. The identification of genes for various disorders was started long ago, but the pace was very, very slow. The most landmark years what I would talk about are the identification of gene for DMD or Duchenne muscular dystrophy, gene for cystic fibrosis, gene for spinal muscular atrophy. However, once the recombinant DNA technology improved, and then even if you don't know the pathogenesis, if you don't know the abnormal protein in that particular disease, one could get identified gene by just positional cloning, and this word replaced the word reverse genetics, which was initially used for positional cloning. Now, with the advent of next generation sequencing, identification of causative genes has become very easy, and this online Mendelian inheritance in man, which is a free catalog of human genes and genetic disorders, has at least 30 to 50 entries per month, as you can see from this record. At present, there are more than 7,000 phenotypes which are single gene disorders, and for some of them, the causative genes are yet to be identified. Another prototype for single gene disorder is achondroplasia, and as contrast to thalassemia, here every patient with achondroplasia has a single mutation. The same base change is there, and hence the diagnosis of based on molecular techniques is very easy. Just it can be possible by 1,000 rupees per patient. However, many other disorders, and I will take an example of other bone disorder or skeletal dysplasia, which is osteogenesis imperfecta, where the causative genes are more than 20, and clinical examination or radiological examination cannot give any clue to the causative gene. And hence sequencing of all these 20 genes one after other by a method called Sanger sequencing, which is shown in this figure, is very, very troublesome, extremely costly, and hence even if we know that the osteogenesis imperfecta needs molecular diagnosis, that testing is extremely difficult. For such cases and for such disorders, there came a powerful technique, next generation sequencing, in this last decade, and it's a very powerful genomic tool for all metamorphosize research as well as patient care. I'm showing here the representative example of how this next generation sequencing is used in patient care or research. This particular picture shows a whole genome and diagrammatic representation, where the coding areas of the gene are represented by a pink color, and they are called exons. In between, there are introns which do not code for the protein, and in addition, there is purple color, lot of repetitive DNA in the human genome. So you can just sequence the coding part of the gene, and this is called exome sequencing, or you can sequence only the genes which are very important for your patient phenotype. Let's say all genes for cardiomyopathy or all genes for retinitis pigmentosa. So such type of high throughput sequencing is possible with this technique called next generation sequencing or NGS, and this has been made diagnosis of monogenic disorders very, very easy. Let us take an example of cardiomyopathy. So cardiomyopathy, again, if you want to find out information about any genetic disorders, go to OMIM, just type OMIM on Google and you will reach this site. So for cardiomyopathy, if you put, there are 872 entries. That means so many genes or phenotypes for cardiomyopathy are reported. If you just see dilated cardiomyopathy, there are 54 causative genes. If you think of hypertrophic cardiomyopathy, there are 34 entries. And if you think of arrhythmias and genetic causes of arrhythmias, there are 14 entries. So naturally, if you have a patient with cardiomyopathy, it is very challenging task if you do not have access to the NGS-based technology. What do you do when you see a patient with cardiomyopathy? Of course, as a cardiologist, you will do the appropriate investigations, but many cardiomyopathies, they are genetic in origin, and hence drawing a pedigree and sometimes relevant investigations and history of family members is very important. And then one can go ahead with the clinical exome sequencing panel, which has genes for all genetic known genetic disorders. Or you can do whole exome sequencing, where it will sequence the sequencing regions of all genes, or you can just go for cardiomyopathy panel, which has at least known genes for cardiomyopathy. And naturally, you may be able to identify some sequence variation in a gene for cardiomyopathy. Sometimes even you can identify more than sequence variations in more than 2 genes. And then there is a question whether this sequence variation is a normal variation seen in normal persons or it's really causing the disease in that particular individual. And this type of things may need family studies, may need information from the literature and lot of databases and so on. Just correlating that the patient has cardiomyopathy and we have found out some sequence variation in a gene which is known to cause cardiomyopathy is not not enough to give make a cause and effect relationship. So American College of Medical Genetics and Genomics have made some guidelines for interpretation of DNA variants. And according to their plan of study, if you identify, you can label a particular genetic variation as likely pathogenic or pathogenic based on the available information about that sequence variation. And these likely pathogenic and pathogenic variants are mostly likely causative variants. However, if the ACMG guidelines decide that this is a benign variant, then you have to ignore it. But the most important and difficult situation is that you cannot make a conclusion whether this is a disease causing or not. And these variants are called variants of uncertain significance. So naturally, every variant in the gene for cardiomyopathy may or may not be causative. And one may get variations in more than one gene, and interpretation in clinical settings is sometimes very, very difficult. This area is evolving. Lot of publications over years are there. I would ask you to go through some such study. And this is a big study of more than 2,500 patients with cardiomyopathy. And they really made a conclusion that 12 genes have robust association with cardiomyopathy. And naturally, for this, you have to go into details. At present, I cannot tell how the conclusion are drawn, but basically you will look at those sequence variations in many normal individuals which are controls, and there are data of many so-called normal individuals in the available, where you can compare the data of your patients and then decide whether this is likely to be causative or not. If the sequence variation or whatever NGS testing is not used appropriately, sometimes it can cause havoc. And that's why I'm discussing this case of 13-year-old boy who died suddenly during sleep. Sudden death being very, very disturbing thing. And then the child had other brother also. The autopsy of the child showed that there was some hypertrophy and diffuse areas of gray-white fibrosis, and definitely type of cardiomyopathy was not labeled. Then the brother was put on investigations, and there was episode of paroxysmal supraventricular tachycardia on monitoring, and they decided that this child has a long QT syndrome, and they gave a defibrillator was implanted. 2 inappropriate shocks were also given. Then because they decided that the boy has long QT syndrome, they did his sequencing by NGS and identified a sequence variation in KCNQ1 gene, which is a gene for long QT syndrome. Then they studied the same sequence variation in many relatives and decided 24 family members have the same disorder, and they ordered or sort of advised them to get defibrillator implanted. Then some of the members came to the Mayo Clinic for second opinion. There, again, everything was restudied. It was found that the variant present in that particular boy who was labeled as long QT syndrome, the variant was not really harmful. Also, the QTC values of that particular child were normal. So they went back to the autopsy blood sample of the boy who had sudden death. So just to make a point that the boy who had sudden death or the disease was not studied by DNA. And when they studied, he did not have this KCK-NQ variant, but again, the exome sequencing was done of that from that sample of the autopsy blood spot, and that was done along with the parents. So you can compare with the DNA sequence of the parents. This helps in interpretation, and whenever you want to order exome sequencing, it is better to order along with the parents if they have normal or if they have, even if they have disease. So this sequence variation is a known sequence variation for cardiomyopathy, dilated cardiomyopathy, and this was de novo, not present in the parents. So naturally, there is no risk to the siblings of the boy who had sudden death. Also, this has to be confirmed by Sanger sequencing, and here on the, I cannot go into the details, but see the number of variants identified in with the whole exome sequencing data. And really to identify the positive variant from such a data is like looking for a needle in the haystack. And naturally here they studied many hundreds of genes for long QT syndrome, Brugada syndrome, etc. for various causes of sudden death, sudden death. And again, then the point again to stress is that unless that particular child who died, unless his investigations give us a final diagnosis or genetic variant, just by studying other relatives affected or unaffected relatives, you cannot make draw any conclusions and make any clinical decisions, especially irreversible decisions. So the messages are that every DNA sequence variation is not pathogenic or causative, and based on variations of uncertain significance, invasive or irreversible decisions should not be taken. The point I again said, the confirmation of the sequence variation in the affected individual is the first step, then only you can go for counseling of the other family members. Now coming to the second part, that in that story, I told you that the parents' sample were also tested and parents do not have any symptoms. But by chance, because you have tested for the whole genome and you find that the mother has a mutation in a gene for breast cancer and father has a mutation for a gene for Parkinsonism, what do you do? Whether you tell them or not tell them, or who decides whether to tell them or not? Which disorders to be looked for, because if it is a treatable disorder like CA breast, you can put the lady on surveillance and improve the outcome, while in case of pre-symptomatic diagnosis of Parkinsonism, there may be issues with job, insurance, etc. So American College of Medical Genetics has given 73 genes, a list of 73 genes, where one should look for such unrelated findings, which are labeled as secondary findings. And these mainly include groups of cancer-related phenotypes, cardiovascular disorders, as I said, because they are actionable and you can do something about them, help the patient, inborn errors of metabolism, etc. So this list has now increased to 73, and it is that is why many cardiologists may get a patient because some doctor has done genetic testing of the child and the parents are found to have some variation in a gene for some cardiovascular disorder, and then they are consulting a cardiologist. So now the question is, who decides? Many parents and patients, individuals may not want to know such secondary findings. And hence, for offering any NGS-based testing, it's very important that enough pre-test, post-test counseling is given. And this is a time-taking thing because they may not understand implications of secondary findings. So this is the list of various genes for which should be told look for in the genetic test if it is done NGS-based. And now the option is given to the families whether they would like to get return of results of such unrelated secondary findings or not. We ourselves have started a small study. At present, the numbers are not adequate, but we are making a questionnaire-based study to find out whether the Indian families or Indian persons, what do they want? They want to know such unexpected results or not. And mostly, I think about 80% or more individuals thought that they would like to get such unrelated findings if they are actionable. Also, when we ask them, do you wish, who should decide? So some people thought that doctor or genetic counselor should decide which results to be given back or not, which should not be given back. So this is a very, very challenging part of the NGS-based testing, and we will have to make some policies for this for Indian population. As I said, the sudden death is a very important cause, and here I show a real case where two siblings from a consanguineous family had deafness and they died suddenly. So deafness with sudden death is a long QT syndrome caused by sequence variations in KCNQ1 gene. And we gave this information, and because the sample of the children was not available, we tested the mother, and she was found to be a carrier of a sequence variation in the gene for long QT with deafness. This was a pathogenic variation. Same was present in heterozygous form in husband as well. So naturally, it must be present in the children, but of course, we did not have the sample. Here again, to point to stress is that like cardiomyopathy, there are hundreds of genes for deafness. And once you test for any individual, you may get variations in 3-4 genes for deafness, and here the same thing happened. But we were interested in a gene for deafness and sudden death, and here we identified the same. Of course, now the important point comes that some carriers of these variants, even in heterozygous form like parents, may have long QT syndrome, and they need evaluation and appropriate management. Lastly, I would just talk about a case with cardiac malformation and issues related to such case. Every case, NGS-based testing is really a research. These two twins had congenital cardiac malformations with the spectrum of tetralogy of Fallot, and we did exome sequencing of both of them. And the sequences showed that they were monozygotic twins, and they had some sequence variations in a gene called PLD1 gene. So there are two sequence variations, one inherited from father and one from the mother. And here I just wanted to show the a screenshot of NGS results, how they see. And you can see that there is a C allele in the, at one point, and then naturally the other is the T. So this 50-50 means the person is heterozygote for this particular variant. Same for the other variant, and this was confirmed that father and mother were carriers of each variant. Now the question is whether these variants which are not present in any databases of normal individuals, whether they are causing disease or not. So we looked at the literature and we found that they were absent in general population, and many of the softwares which can say whether this particular sequence variation will likely to be damaging or not, or then hence causing the disease. So most of them showed that it was disease causing. But according to criteria of American ACMG, they come to be variations of uncertain significance. So we again go back to the OMIM and then find out that this is particular gene, PLD1 gene, causes valvular defects and structural malformations. And they are also in the spectrum of the same tetralogy of Fallot. There usually, as I said, OMIM is very up to date, but here you can see for this particular gene, the entries are only up to 2017. And these papers, what they showed that these three cases from two families had tricuspid valotresia, pulmonary valotresia, VSD, and almost similar spectrum like the children in our family. So our children were operated and they were doing fine. So we decided that this is likely to be causing the disorder in that particular family. So look, now we were thinking that this could be disease causing. That was 2 years ago or 3 years ago. And now while I was making this PowerPoint presentation, I again yesterday looked at that particular gene and see how fast the information gets updated. There is a paper in 2021 on the 2,700 patients of congenital heart disease, and 21 of them had mutations in these causative mutations in the PLD1 gene, and 4 of them had this mutation at the same point, but other than A to T, our patient had A to G, and hence the amino acid also change was different. But this gives a very supporting evidence that this is the causative variant in that particular family. Lastly, there is a talk about these multifactorial disorders, but I would just, for the sake of completion, would mention that, as I said, whether it is diabetes, hypertension, ischemic heart disease, psychiatric illness, everybody has genetic etiology and variant forms. And these individuals, some of the individuals have more variations in the genetic susceptibility of these disorders. And these genetic variations can be looked for to find out whether they have high risk of a particular disorder, multifactorial disorder. But the point to mention is that in addition to those genetic variations, environmental factors are also important for multifactorial disorders. And the difference between the monogenic disorders and multifactorial disorders is that for monogenic disorders, there will be a sequence variation only in one gene. And if that pathogenic sequence variation is in the cystic fibrosis gene, the patient will always have cystic fibrosis. While for multifactorial disorders, there are variations in many genes, and which these variations also may be present in normal individuals. As you can see, the genetic variation which gives susceptibility to type 1 diabetes patients may also be present in normals, but in a lower frequency. So naturally, presence or absence of that sequence variation does not give you definite idea that whether the individual will have a disease or not. And also the most important thing is what will be the age of presentation. So these sequence variations study for multifactorial disorders are very important because they give insights into the pathogenesis of the disorders, and also probably may help in development of some treatments for them. But polygenic risk scores are just give probabilities and not certainties. And there are no guidelines for practice of these particular polygenic risk scores. They are associations and not causal relationships. So sometimes I like to compare them something like the results of various predictions based on Rashi Patrika, that possibility of early death and so on. And lastly, the even interventions, if you see that there is a high risk of a particular disorder, the interventions are mainly lifestyle modifications, and probably the same information can be obtained based on the family history, BMI, and lipid profile. So ACMG again has a position statement on the use of these direct-to-consumer testing for multigenic or polygenic disorders. And what they say that the genetic studies on complex traits and disease is still an inexact science, and do not identify affected individuals as the testing for monogenic disorders does. It means that probably it will give some possibility, but not tell definitely. And secondly, they say that ACMG, it is not a part of general clinical genetic practice. And in the context, the emergence of direct-to-consumer testing for polygenic disorders is particularly troubling, as according to ACMG. Rather than if good clinical information it can be greatly of use if you see a child, individual with cardiomyopathy and identify these angiokeratomas, and then you can make a diagnosis of Fabry disease. So at the end, I will say that how powerful the genomic techniques may be there, clinical examination and clinical skills remain an important part of patient diagnosis. So at this era, when everybody wants to be empowered with the genomic information, I request all of you to join our Society of Indian Academy of Medical Genetics. Thank you very much.
Hello.
Yeah.
Hello. This is Shubha Farke.
Yeah, have you joined us?
Yeah, I'm there. And can I answer the previous discussion which was going after?
You can, please.
Because that is going to be most important question.
Yeah,
the first simplest answer is for any patient where monogenic disorder is suspected, you should go ahead with whole exome sequencing. The reason, as I mentioned in the previous case, I highlighted that if you do a panel for cardiomyopathy, so the panel may be decided or devised 1 year ago or 1 month ago, but it may not have some important genetic gene which is recently identified in last 6 months or last 1 year. And hence, if you do a panel, it may not cover a very important relevant gene which may be present in your patient. Of course, in how many percentage of cases this will miss because you have used panel and not exome cannot be exactly calculated. Secondly, previously the panels were being done because we thought that they give good coverage because they are testing for only few genes. But now the whole exome sequencing techniques are so better that the coverage of all genes included is very good. And regarding the cost, as compared to panel versus the whole exome sequencing, the cost usually most of the laboratories have 3,000 to 5,000 rupees more than panel. So I don't think that panel testing has a very important role, except something like that. I know that this is Duchenne muscular dystrophy only, but I'm doing NGS-based testing because this gene is very big. Then I may order a whole panel for DMD or panel for muscular dystrophies. But otherwise, whole exome sequencing should be done for all. If possible, affected and unaffected parents in the family member may be added, so trio sequencing adds. But here in case of cardiomyopathy, then both the parents should have undergone appropriate evaluation to find out whether they have cardiac involvement or not. So hope I answered the question.
Well, thank you so much, madam. That was clearer. Only thing is that we leave that to as cardiologists. Sorry, somebody else can mute themselves here. Yeah. So as cardiologists, we should be knowledgeable enough to decide about the panel, or we should leave it. And how do we really figure out how up to date is that particular lab?
So that is why I'm saying only go for whole exome sequencing, because that will have most of the genes which are not even at present labeled as cause for cardiomyopathy, and they will be there. So once you get that sequence variation, then you can look into the literature and find out if it is associated with a cardiomyopathy or not. And so that brings me to the other important point to highlight is that now everything, even 15, 20 years ago, some visionary had written that all investigations gradually will become automated. That is really happened. And then the clinicians' input regarding the interpretation of the results, like the lab has given you some interpretation, these are likely candidate variants. Then cardiologist or any clinician can give input in details by looking at the patient, looking at the literature about that particular gene and genetic variant. So clinical collaboration is very important. I do not mean that just correlate clinically that cardiomyopathy is there and the patient has sequence variants in cardiomyopathy gene. Such type of correlation is very weak. That evidence is not considered very high. But if you feel that the patient has cardiomyopathy and Fabry gene variant has been identified, then you go and look for angiokeratomas and also do enzyme for a Fabry disease. And then it is also low, then it gives enough supporting evidence. Regarding VOUS, there will be always this VOUS daemon will be there always in front of us. But you have to deal with that according to the situation. As far as, sorry for the background noise, So you have to deal with appropriately. First important thing is don't do any invasive test procedures based on the VOUS. Don't do prenatal diagnosis based on the VOUS. But if you do say pre-symptomatic diagnosis and find out that the son has also same variant, then at least you can keep on surveillance of that particular child and see if that they develop disease or not. So VOUS, and I understand because most of the variations will be VOUS because we do not have a lot of data about the cardiomyopathy variants in Indian population. That is one limitation. Second is we do not have data of variations in Indian normal population. That is also limited. So naturally, if you find out some variant in cardiomyopathy gene, but that may be present in many normal individuals in India, but not yet reported. So hence, these are the limitations. And of course, naturally, so whenever you order the patient, you give enough time to the patient to explain. When you get the result, you take 8 days for yourself to learn about that sequence variation. You can learn to study and use the various databases of normal variations as well as disease-causing variations. You can consult a geneticist and then probably give time to the appointment to the patient and talk to him about that variant so that one feels reasonably confident. I say reasonably in the sense because there will be some situations where you have to explain to the patient that there is uncertainty and Are Baba, I cannot do about this uncertainty. So we'll have to wait and watch and a lot of things.
Yeah, that's Indian versus non-Indian becomes quite important, isn't it? So Indian VUSs, we do not have too many, which reminds me in the study that we collaborated with for hypertrophic cardiomyopathy, 100 odd patient samples with controls also. And we did find out 2 VUSs out there in that particular study, but then unless we have an Indian data, how does this VUSs, how often in the world literature do VUSs tend to become benign afterwards?
Everywhere in the world, there will be VOUS because there are so many sequence variations and there are the 3 billion base pairs. So as more and more information about each nucleotide, as it gets annotated, reported in normal individual and reported. So whole world is having lots of bioinformatics work going on this, and nobody can say percentage of VOS which will become, there are people, CleanVar is a database and people have seen that in the entries in that CleanVar databases reported as VOS, how many of them have become, but that would be vary depending on the patient disease and so on. So basically the dictum is VOS, that can be like if you feel heart in heart, this patient has the same phenotype. So you may think that this is likely causative, but will not take any irreversible decisions based on that. So this happens, like I have a patient with immunodeficiency and absent nails, and I get a sequence variation in a gene which causes immunodeficiency and absent nails. So this is a very, very specific phenotype. And then I will say as clinically, I will take decision that this is a VOUS, but based on that, I will convey the limitations to the family and I may do prenatal diagnosis. But if it is only just immunodeficiency, then there are hundreds of genes for immunodeficiency. So I will not do prenatal diagnosis based on the very non-specific phenotype of immunodeficiency and some other findings. So this type of clinical correlation and getting data is important. And sometimes we have seen that over years, as we evolve, we really get really some other variations. So in Indian population also, we ourselves as a SIMG and under ICM are trying to make a databases of disease-causing mutations in India, and also variations in, you can see one paper published by from Manipal group about variations in normal population in India. That data is freely available on the website of the CDFD database. So lot of things are there, but of course it's a huge task. And
well, thank you, madam. In the interest of time, I think we need to move on.
Thank you.
And we will come back again to you. That's an excellent talk. Thank you so much. My co-chairs had some questions to ask, or whether you want to go ahead.
We can, madam, you are going to be with us throughout the program. So we will.
Yes, yes, I will try to do, but as you can understand, I've just, because of the change of date, I'm traveling somewhere. and some
Right.
problems with the connectivity may be there, but I will try to be
No, because
try
your
to
voice,
remain
your
attached.
audio and video both are excellent, better than what we are having ourselves here.
So,
I think
just
you are
to
doing
tell,
well, madam.
I'm in Krishna's Dwarka, on the bed Dwarka.
Wow.
So
God is with
that
you.
is
So
the
that's why
Jai
we are
Shri
development
getting
in
a good
Krishna.
India.
transmission.
Thanks
So
a lot.
please be with us throughout the program, because your
Thank you.
inputs will be the
Thank you.
most important for us. Yeah, thank you.
Jai,
Praful, can you go ahead?
Jai Shri Krishna, yeah.
Praful, just announce the next.
Yeah, Praful, carry
Hello,
on.
my good friend, Dr. Rajiv Gupta. We've heard about your CV already. We don't need to introduce us to Mumbai crowd. But that is the very exciting area, isn't it? We all talk about young CAD, and what is the geneticist done for us so far? And give us an overview of all the polygenicity about it. We have the famous Dr. Rajiv Gupta. Go ahead, sir.
Thank you, Praful, and thank you, friends from Mumbai, for inviting me here. Dr. Shobha Fatke has bored me with a googly, actually, so she has already done that. But I'm going to talk to you. Have you started the share screen, please?
Yeah, please share the screen. We can't yet. We are still on a gallery view.
Yeah.
Great. We can see it. Go on a slideshow.
Yeah. So thank you very much. And what I'm going to do today is to just calculate the risk score and can we predict heart disease. As cardiologists, we are all trained to use incomplete information about the patient and to make decisions. And this is just a part of that particular sequence. Now, I began with a premature heart disease, and you see here is a 39-year-old businessman who came to me with acute coronary event almost 20 years ago, inferior wall MI, heart block, thrombolized. That time there was no fashion of primary PCIs, no clinical risk factor excepting family history. And the coronary angiography showed a disease in the LAD and the right coronary, and he had a bare metal stent in both the arteries. Of course, standard pharmacotherapy, and he's doing well. So the question which comes to mind is, why did it happen and what are the risk factors? Now, these are latest data from the PURE study on the risk factors in South Asians. And you clearly see that they are all old people risk factors like hypertension, non-HDL cholesterol, pollution, smoking, diabetes, and diet. So all these things are well known, nothing new here. And we also did a study on premature heart disease. They are less than 40 years of age. And again, we find that smoking was the most important risk factor. Of course, homocysteine, HDL, hypertension, triglycerides, and everything else we all know on premature heart disease. So what do we do in these patients? Can we predict the risk of heart attack in this patient and the patient that we see less than 40 years or 50 years of age in our clinics? We should remember that heart disease is multifactorial, we all know that. And there are various risk prediction tools. Now, we all know of Framingham and Reynolds and the all score, a sign score, QRISK, all based on Caucasian populations. Then there are certain international risk scores like the World Health Organization risk score, WHO. Global is a global collaboration. It is done by the Non-Communicable Disease Risk Factor Collaboration from UK. And they have had two variations of that. InterHeart study also had a risk score. And of course, now we have bioimaging like coronary calcium scores, but they are basically atherosclerosis, mild atherosclerosis in a particular patient. So bioimaging risk is not really a risk predictive score. I put all these risk scores in our patient. And what do we find? Framingham risk score, the 10-year risk in this particular patient was only less than 1%. Pooled cohort equation, again, less than 1%. And the QRISK-3, if we put in the South Asian, it comes to 2.8. And WHO, InterHeart, and global risk between 5 to 8%. So again, clearly showing that none of these risk factors, risk scores at least predicted heart disease in this particular patient, especially in all our young patients. That's not only true for Indian patients. Now, this is a study from the Harvard, the Young MI Survivor Study, Partners Group, Harvard. And what they asked is adults less than 50 years of age with first-time MI, were they eligible for statins? And what they found was only half of them were eligible and half of them had no risk factors. Again, showing that it's very difficult to predict premature heart disease in using conventional risk scores. Now, we published a couple of years ago, an article in the Lancet on the primary prevention of heart disease. And there we sort of made this particular cartoon or diagram. So we all know that there are individual risk factors, individual level, lifestyle variations, behaviors. But what I presumed was that genes are important and genetic risk factors have a contribution to all of them. How do we find these genes? And in the previous two talks, there have been talks on linkage studies, on the family-based association studies, in the candidate gene association studies, genome-wide association studies, and deep sequencing. All of them have their good things and bad things. Linkage studies were the old ones. You need large pedigrees, relatives, affected SIP pairs. That's what Professor Fatke was talking about. Then again, family-based association studies, you have parent-child trials. They are basically good for single gene disorders or poly-gene disorders, as you call them. And then you have unrelated individuals, which has large sort of multifactorial disorders. Hello? Yeah. In these multifactorial disorders, you have candidate gene association studies, genome-wide association studies, and the deep sequencing studies. You can simultaneously study multiple genes across the exome or across the genome, and then you can find out certain areas of interest and what are the important genes as compared to the control group. This is just an example of showing that whole exome sequencing. There are genes in the Caucasians and there are certain genes also in the South Asians given in the red. So these are the South Asian genes, the blue are the Caucasian genes, especially for coronary artery disease. A rather complex area, but just to show that there is a huge amount of variation as far as the coronary artery disease is concerned. So in the year 2011, when it all began, actually, it began with the Human Genome Project. But then what we found was that using genome-wide sequencing, you identified 42 genes, which were in a case control study design, and these were the most significant, especially the 9p21 genes, and then other areas. And they also found that there are certain genes in South Asians which were important. And again, you see lipids and ADAMs, this blood group genes and other genes being important. So that's where the concept of the gene risk score developed. Now, this was because as we know that this is not a multifactorial disease, this is not a single gene disease, it's a multifactorial disease, and every gene which has effect has hundreds and thousands of polymorphisms. So you could have a large number of genetic variation to explain the risk. So we came from the single gene etiology to the polygene etiology. That means the risk factor development. And now, since the last 10 years or so, people are not talking of atherosclerosis as a single gene disease. They're talking of atherosclerosis as a risk. And we all know that just like hypertension, just like high cholesterol, just like BMI, there are certain group of people who carry this high gene, polygenes, and they are at the high risk. One of the first papers was from Finland, and in the Finn risk study, they studied 13 recently discovered single nucleotide polymorphisms or single nucleotide variants, as they are known now, and association with coronary artery disease. And what they found was that as the number of genes increased, That means from one single as a low quintile to the highest quintile, the fifth quintile, the risk went up from 1 to 1.7. So it was a 1.7 time predictive of coronary artery disease, about 1.4 times predictive of acute myocardial infarction. Now, this is something like smoking. If you smoke, the risk goes up to about 1.5 to 2 times. And the same thing is for genes, actually. If you have a higher gene risk score, the risk of the subsequent heart attacks go up. Now, this was for the whole population. This is not only in premature heart disease. So I think as we go along, we'll realize that in premature heart disease, they are much more important. Then they talked of 27 gene variants. This is a study from US, a combination of 4 studies among women and men. And what they found again was that as the gene risk score goes up, the risk goes up to 1.72. So that means people at the highest end of the risk have a 70% greater chances of developing acute coronary syndrome or coronary artery disease as compared to the low risk individuals. Again, showing that there are the risk factors of association, like smoking, like hypertension, which provide the same kind of relative risk. Then they went on from 27 to 50 gene variations, and again they found out the same type. This is from Europe, and they thought that as compared to the 27 gene risk score, if you have a 50 gene risk score, you can predict better. So from 1.7 it went to 1.92 in the highest gene risk score category. Some more genes were added, 60 gene risk score for CAD prediction in 2016, and again showing the same thing, that 1.7 times or 1. And this is particularly important because this is the ERIC study, the atherosclerosis risk in communities, basically a mixture of black and white individuals, and again showing the 1.75 hazard ratio. But this is women's genome health study, and what they found here was the risk was even much more in women, 1.94 times predictive. So one area which I think is important to further investigate or further evaluate is that do gene risk score predict greater risk in women as compared to men? We don't have a large database from India as I understand. Studies have also shown that the gene risk score are better than family history. So clearly, in a premature heart disease, if you just have family history, you may have, say, 40% greater risk. But you combine that with the gene risk score, the risk goes up to about 80%. So clearly, you can double the predictive power of the family history by adding the gene risk score. It's even better than familial hypercholesterolemia, which we all know that is a very important factor for coronary atherosclerosis. Then we come to 49,000 SNPs. Again, I think as the times are changing, we are increasing number of the SN single nucleotide polymorphisms in the gene risk score. Again, combining 3 or 4 studies, and again showing that this is the Finland's risk study in men, 80 to 100% greater risk. This is the Framingham Heart study in men, about 40%. In women also a slightly higher amount of risk. So these things are evolving, and what you find is that multiple studies, this is the World Wellcome Trust Case Control Consortium, MI-GEN study, Argos, FinRisk, and Framingham Heart. Again, all of them showing that the risk of this 49,000 gene risk score in the prediction of heart disease as compared to low risk individual to the high risk individual as compared to cases and controls is between 28 to 75%. And I call that in Caucasian, the risk is about 1.5 to 1.6 times if you have low gene risk score versus the high gene risk score. Genes are evolving. Now we know there are multiple genes across the board because we have multiple risk factors like LDL, triglyceride, inflammation, proliferation, thrombosis. And you have multiple genes for each of these processes of atherosclerosis. And remember, every gene has hundreds and thousands of variations. So clearly, you're not dealing with 29 gene or 30 gene, you are dealing with multiple variations, multiple SNPs for each of these genes. And that's why they are evolving as far as the gene risk score is concerned. We began from 50 to 50,000 to 115,000, and now we have 6 million as single nucleotide variations in a particular gene risk score. So they identify individuals with high risk, which is equivalent to monogenic mutations. And what you see clearly here is that if you increase the number of genes, so this is 50 variations, you can predict about 6% of disease. In 50,000 variations, 8%. And now with the genome-wide PRS, 6.6 million numbers, and you can have a very large amount of prediction. You keep on increasing the predictive capabilities using all these gene risk scores. So I come to South Asia and India because that's very important. And what this particular article showed was there was a promise of discovering population-specific disease-associated genes in South Asia. And this is a study from CCMB Hyderabad and multiple collaborators across the globe. And what they found was that you could do a whole genome sequencing in these particular areas. And New York Times published a very interesting article on that particular paper, because in South Asia, we have social castes. And what New York Times columnists believe that this could be a living lab because every social caste varies within themselves. So within this particular social caste, there could be an increase in, for example, cardiomyopathies, for example, arrhythmic channelopathies, for example, atherosclerosis. So that's what they predicted that this should happen in India, and that's what's happening. A study from Pakistan, a very small study, 625, 405 cases, 220 controls. And again, you see, they only have 21 variants and 18 genes, and the highest risk is 3 times. Remember, in Caucasians, it was 1.5. In Pakistan, it is 3 times the greater risk, 2.96. Again, showing that in South Asia, maybe we are more prone to develop that. So we performed a study, multi-site study in India. So there were 4 sites, Chennai, Bangalore, Jaipur, and one more in South India, Mysore. And we did a case control study, premature heart disease, less than 55 years of age, the standard definition. And what we tried to do is to validate the genome-wide risk score for CAD in South Asians. And we compared the data of Indian data with 2 groups, 2 cohorts. One was the Bangladesh heart study known as BRAVE, and the other was the South Asian in the UK Biobank, which is again South Asian settled in the UK base. And it's a mixed bag in the UK because they are all North Indians and South Indians and East Indians and West Indians. So they all have combined together. And what they clearly found was that the risk score predicted in the UK Biobank, the same degree of prediction, about 3 to 4 times. That is, the odds ratio was 3.5 to 4. And the same thing happened in Bangladesh as the risk score that we developed in our cohort, in our using the South Asian genotypes, again predicted the Bangladeshi also in the same sort of setup. So this is the, so I just trying to show you that in South Asians, the genetic factors maybe are more important. And you see the relative risk in, at least in the UK Biobank, about 8,000 patients of incident myocardial infarction. If you have the highest quintile, the risk is, odd ratio is 5.5. So clearly showing that genes are very important in our population, especially with premature coronary artery disease. In Bangladesh, it was 2.4. And what happened in our own cohort in India, 1,800 coronary artery disease patients, 1163 controls, percentile of high risk, genome-wide risk score was high. And again, you can see clearly here is that if the gene risk score increased, the odd ratio kept on going up. And at the highest end of the gene risk score, it was almost 5 times the risk. So clearly showing that in patients with premature coronary artery disease, you can predict the occurrence of premature coronary artery disease using this particular risk score. We need to validate. So we, in this particular study, a genome-wide risk score to identify patients at risk of coronary artery disease. It integrates information from common DNA variants into a single measure. So that's why we did a South Asian, but we need additional research to improve the transferability of genome-wide risk score across various racial and ethnic groups and facilitate their integration into clinical practice. Can we do that? We go back to our patient, and we have already seen that the classical risk prediction scores only work up to a certain level. But in our patient, if we applied the polygenic risk score in our patient, and there was taking, he was in the 95th percentile, so at a very high percentile, between 85 to 90 percentile, and which corresponds to about 20% 10-year risk. So clearly, the gene risk score did better, much better as compared to the standard risk estimation scores across the globe. So clearly, there is something there. Of course, we need more studies. We need more prospective studies. Case control studies are not the best way of identifying all this, but then we have something to predict disease in premature coronary artery disease. Can the PRS predict MI in the young? Now here you have data from the UK Biobank, less than 50 years of age, incident MI, using different kinds of score. And you see clearly the receivable operating curves. If you use a very large 1.5 million, 6 million S&P based risk estimation score. This is the red line. And you see clearly it is a very good predictor of premature myocardial infarction. So clearly premature heart disease can be predicted by gene risk score. Again, you see here the risk score in the young. Again, another study in the UK Biobank, and the best prediction was in 40 to 49 years. So there's some typing mistake. In the earlier age, so premature heart disease, there was a 2 times odd ratio prediction, both in men and women. Women was 1.57, and men is 2. So clearly premature heart disease is predicted much better by gene risk score as compared to heart disease occurring at a later life. An important question is, can we predict second heart attacks? Suppose we have a patient with a premature heart disease, and can the risk score predict recurrent events after myocardial infarction in the young? And this is a study from Spain, a very small study of 81 patients. They determined the gene risk score with 11 variants, and the follow-up was about 8 years. And what they found was that as compared to people with the low gene risk scores and the higher gene risk score, highest gene risk score predicted much more events as compared to the lower gene risk score. Now this has clinical implications. That means if you see a young patient with a high gene risk score, you can give him more aggressive therapies. And we all know what therapies are, like statins, like lifestyle changes, like antiplatelet drugs. We need more trials on that, clearly, but then there is some information that secondary heart attacks in a patient with premature coronary artery disease can be usefully predicted by the gene risk score. And finally, what is the ideal risk score for Indians? Now, there are 2 types of risk scores. There are clinical risk scores and there are gene risk scores now. The clinical risk scores are well validated in Caucasians. They are widely used in practice. They are more applicable in middle age. And unfortunately, the biggest drawback of clinical risk score is that age is the most important risk factor. And as the age increases, the risk increases. using these clinical risk scores. And of course, for India, inter-heart, global risk 2 and British risk 3 may be useful, but we don't have data from our country on prospective studies. Gene risk scores are clinically appealing. Of course, they are only validated in Caucasians, excepting the study that we did in our country, a small study, which is still ongoing. Fortunately, it is applicable at young age. We need studies, many more studies from our country, and the costs are rapidly declining. So it's not the issue of cost, it's basically issue of the science behind the whole thing. Eric Topol published an article in some years ago in Nature Reviews, and he's just trying to find out what can we do, clinical risk score versus gene risk score, and you have or the combined risk score. So what he suggested is that maybe combined risk score, combining both clinical risk and the gene risk, is the best thing. And you have an action threshold that beyond this particular thing, at least for primary prevention and as well as for secondary prevention, you can start aggressive risk factor management. So can we do what interventions? So what are the interventions? And if I talk to patients, say that we want to do a gene risk score, most of the patients refuse. They say, Saab, what will we do? Kya hoga? Fayda kya hoga humko? If we identify us as a high risk, will it help us? Two interventions are useful. One is the lifestyle, and there's been a trial from US, 3 different kinds of studies, ERIC, women's health, and the MELMO, which is a European study. And all clearly show that people who are having a favorable lifestyle reduce the high gene risk. So clearly, a good lifestyle protects even if you have a high gene risk score. So clearly, lifestyle is useful. And here you have a trial on statins. Just again, a combination of 4 primary prevention trials, Jupiter, Escort, CARE, and PROVE-IT 22. PROVE-IT is, of course, a secondary prevention study. And again, showing that statins attenuate the gene risk. So clearly, you have 2 interventions. If you have a high gene risk score, you can prescribe good lifestyle, and you can prescribe statins. So friends, we all know that premature heart disease is a major problem. Traditional risk scoring systems are not useful, especially in the young. The gene studies have identified numerous SNPs as important. We now have gene risk, GWAS-based gene risk scores with more than 6 million genes, 6 million SNPs, it's not genes actually, that have been developed and assessed in the US and India. They provide robust risk assessment, and they add to precision medicine, especially for prevention of premature coronary artery disease. Thank you.
Thank you, Dr. Gupta. Dr. Kher, permit me 5 minutes delayed, so we will straight move on to the next session and take up any discussion at the end of it. And I would request all the chairpersons of the first session also remain along with the other chairpersons so that we can have a combined discussion at the end of all the talks. I would now invite the chairpersons for the second, Dr. Akshay Mehta, Dr. Karnik, Dr. Burkul to take over. Dr. Karnik, could you take over from here, please?
Yes, thank you very much, GRK. Am I audible? Am I audible to everybody?
Yes, you are. Yeah.
Dr.
Gupta, be with us. We'll take up the discussion at
thank you very much, Dr. Kane, for involving us, me and Akshay and Nitin Burkule. Ajit was also there, but then I think he's backing up the most important person of the state. So I think he would not join us. So 3 of us would be in the chairing position. Now, we had really excellent session, and Dr. Kane and the Praful and Sadanand, I mean, really marvelous, innovative talk. I think very novel talk, because so far CSI did not have any meeting on genetics as such. Now, the first scintillating, the most important talk is going to be by Dr. Amit Vora, and it is about channelopathy and with genetics in focus. Dear friends, all of us, whenever any patient with sudden cardiac death or ventricular arrhythmia or syncopal VT comes, we have 7 different steps in which we do the investigation. And G comes second, and G is genetic testing. So A, B, C, D, E, F, G. And same thing happens in our clinical practice. E is ECG, second is electrolyte, third is echocardiography bedside, fourth is EP study, coronary angiography, cardiac MR, 6 are over, and 7th is generally the genetic testing, if at all, if any one of us does. But then I think today after Dr. Amit Vora's talk, maybe, and then after this scintillating earlier, the very illuminative talks by 3 giants about genetics in cardiology, I think we may escalate the position of this genetic testing upward. So with this little short introduction, let me invite Dr. Amit Vora for channelopathies and with genetic testing in focus.
Thank you. Thank you, Rajiv, and good afternoon to all of you. I hope my screen is also visible and I'm audible.
Yes, you are.
All right, thank you. So in terms of channelopathy causing sudden cardiac deaths, I think as a clinician, we need to remember of long QT syndrome, Brugada syndrome, catecholaminergic polymorphic ventricular tachycardia, arrhythmogenic RV cardiomyopathy, and short QT syndrome. Now, the genes which are responsible for these syndromes also is important for us to understand. In long QT syndrome, there are probably more than 11 syndromes described, and there are more than 30 genes. But as far as the genes which have become absolutely standard and validated and most important, there are only the 3 genes which we need to remember: the KCNQ1, KCNH2, and SCN5A. For short QT syndrome, they are the similar genes. In Brugada syndrome, it is SCN5A, and in catecholaminergic polymorphic ventricular tachycardia, it is essentially RYR2, and less common, the CASQ2. So I think it's just important for us to remember these genes when we are ordering the genetic studies. Now, let me move on to what is more important in terms of our clinical experience with the disease and the genetic studies. And I'll share 2 short cases. The first case is of a 13-year-old girl who presented in 2016 to me with exertional syncope. And the story was that she had a twin sister who had a sudden death while playing a sport. So with this background, when she came and was referred, the ECG done, this was of 2016, but yet the ECG clearly shows that you can hardly pinpoint any abnormality. If you look at the QT interval, the corrected QT is 420 milliseconds. There is no evidence of any arrhythmogenic RV cardiomyopathy or a Brugada kind of a pattern. And there was no chamber enlargement also to suggest HCM or any other cardiomyopathies. So obviously, in terms of the investigations, we definitely want to rule out any myopathy. So echocardiogram revealed a structurally and functionally normal heart. Holter was done. On the Holter, no arrhythmias were picked up, but there was some suggestion of a maladaptation of QT interval. Now, QT interval adapts to changes in heart rate. And if you have some maladaptation, so at a higher heart rate, you would expect the QT to shorten. But if it doesn't proportionately shorten, then you call it maladaptation. So that was what was noted in the Holter, but overall the QT was normal. It's always important to do ECG of the parents when you have these kind of sudden deaths in the family, and the ECG of the parents was normal. So I think we were still in a flux based on these investigations. We have exertional syncope as a clinical pointer, but the ECG echo more or less normal. So exertional syncope, long QT can be a problem. CPVT could be a potential problem, but we are not definitely ruling out any form of myopathy so far. And we know the value of cardiac MR. It is much more sensitive in terms of picking up structural abnormalities and certain myopathies. So we did a cardiac MR in this girl, and that also was normal. And essentially, we were trying to look for arrhythmogenic RV cardiomyopathy and hypertrophic cardiomyopathy. So this was normal. So now this child had a exertional syncope. So we planned to do a stress test, and we saw some maladaptation of QT interval also. So during the stress test, actually, we found that the QT interval behaved very well and there was no problem. However, we had a non-sustained polymorphic PT, which was just for a few seconds, and we terminated the test, and with rest, again, everything normalized. So my provisional working diagnosis was that we are dealing with catecholaminergic polymorphic ventricular tachycardia. I think the classic ECG presentation is of a bidirectional VT, but you could have a polymorphic VT, exertional syncope, sudden death in the family with echo MR being normal. So this was my working diagnosis. What did I advise? I advised propranolol 5 milligram per kg and per day, and we generally step it up to 10 milligram per kg per day. I did suggest them to consider a stellate ganglionectomy. Now, stellate ganglionectomy works very well for long QT and for CPVT. Yet the diagnosis was in an uncertain territory, and therefore genetic testing for confirmation of the diagnosis was advised. What did I get in terms of the genetic testing? We did get the RYR2 gene positive in terms of it being possibly a catecholaminergic polymorphic ventricular tachycardia. It's of autosomal dominant inheritance, and the classification given was likely pathogenic. So this is likely pathogenic. So this is probably disease causing. It also fitted into the clinical provisional diagnosis. So I think there was some benefit in terms of doing this genetic study. But what was also confusing to us was this variant of uncertain significance. What also was found is a PKP2 gene, which predicts arrhythmogenic RV cardiomyopathy or dysplasia, but this gene is of a uncertain significance. So this becomes a challenge, that what is still the true diagnosis. I think based on the clinical presentation, based on the stress test and everything, we would continue favoring CPVT as a diagnosis, but this throws up some kind of questions. But we know that when you have uncertain significance, you should not direct your tests or treatment based on that. And therefore, we continue to treating the child as CPVT. But what we decided is that let's do the genetic study of the parents, because that might further help. So father and mother, both genetic testing was done, but they are asymptomatic, and the genetic testing for RYR2 exon came negative. So this was absent. So again, now we are in a bit of a dilemma. Next gen sequencing for YR2 in the family, at least the parents, was negative. Yet my working diagnosis continue to be in terms of CPVT. So to give you a follow-up of this patient, while on propranolol, the child has had no syncopal spells, but yes, the parents did report that she at times misses few doses of the drug, and at those periods, those days, she has had syncopal event. And therefore, I stressed the need for undergoing stellate ganglionectomy, because we have a malignant family history of sudden cardiac death in her sister. On propranolol, has been 4 years free of symptoms, and things were otherwise going well till just last month. On propranolol, apparently this time the drug was not omitted, and the child had another syncopal spell. So this is the time the event monitor was done. They are from Solapur area. The cardiologist who had referred me the case did an event monitor there, and an event monitor, again, a non-sustained ventricular tachycardia of polymorphic nature was picked up. So we are thinking that this is CPVT. Beta blocker probably is not sufficing, though the dose has been stepped up. But most important is that we have emphasized the need for getting a stellate ganglionectomy. And actually, she is coming to Mumbai for a stellate ganglionectomy in the coming week. So I guess as far as this case is concerned, there are two issues which we could discuss. Is there a role for repeat cardiac MR and ruling out arrhythmogenic RV cardiomyopathy? Because we know that often the clinical presentation precedes the ECG or echocardiographic findings, and whether I should do a follow-up cardiac MR 5-6 years down the road to make certain that that continues to remain normal. We are not dealing with ARVC, and we have CPVT only as a diagnosis. And of course, when you have a malignant event, the question always comes up of an ICD implantation, whether I should do that or not. So I think I'll pause here and go to the next case, which is going to be very brief or short, and then we can have the panelists and the discussions opened up. So this is a short case relatively. Again, a young 7-year-old girl who had a single episode of syncope. And historically, when I go into the depth of the story, I have a strong suspicion that it was a vasovagal phenomenon. Now, this patient has an interesting reference to me because there is a family history of Marfan syndrome. And her uncle had aortic aneurysm, aortic regurgitation, had required aortic root and ascending aorta replacement surgery done. And that also had become aneurysmal, and a repeat surgery actually was done a few months ago. Unfortunately, had a very stormy course post-operatively, bled and died at a young age of 45 years. So the child's father, who is the brother of the person who died, father also has Marfan's features. All Marfan clinical features are there, but father is asymptomatic. Girl actually also has Marfan features, and the girl's ECG is normal. The echocardiogram showed a tricuspid valve prolapse with a mild TR, but otherwise everything was okay. So a whole exome sequencing was done in this child to be able to identify what was the problem. And they found this gene SBN1, which predicts the likelihood of Marfan syndrome in this patient also. But unfortunately, the classification given is uncertain significance. Now, this is the problem, that the targeted gene sequence not done and a whole exome sequencing done, it throws up many other diagnoses also. So what it also additionally showed is RYR2 gene, again of uncertain significance. This predicts ARVD and CPVT. And it also showed an HSE gene, which is pathogenic, but with a reduced penetrance, and this predicts hemochromatosis. So when we have so much information coming from the genetic studies, it becomes challenging as a clinician to decide what should we do. And actually, I was referred by the geneticist and the pediatrician because of this RYR2, and they said the child has had one episode of syncope and RYR2. Would you think in terms of CPVT or any channelopathy which could cause sudden death? And that is where I, looking at the clinical profile of the child, strongly thought that the history was of vasovagal syncope, not related. So RYR2 variant of uncertain significance probably has no value, and I would not really like to chase it any further. Having said that, a simple test always could be done. So I did a 3-day event monitor, and the aim was basically to pick up arrhythmias, and especially when the child has a, with activity as sinus tachycardia are there any PVCs or non-sustained VT coming up? And you have a heart rate of 177 in the afternoon when she does some exercise or playing some sport, and you have sinus tachycardia, no arrhythmia whatsoever. At the minimum heart rate had been 50 at early morning hours, and again normal ECG. So I have stopped further investigations in her and told them that the RYR2 gene in her case of uncertain significance need not be chased any further at this point in time. But I don't know whether I'm completely right, and so that's where it comes. What is the significance of genetic testing? The previous speaker has spoken to us, but this is what we need to understand, that some of them are pathogenic, which are likely the causative of the clinical problem. Some of them are pathogenic with reduced penetrance. What to make of it, likely pathogenic, again, are of importance. Uncertain significance is a borderline category, could go either way, and we probably need to correlate with clinical situation and follow up. Benign are definitely not related. And we also need to understand this concept in genetic testing as secondary or incidental genetic finding. And I think in the second patient, I think the presence of RYR2 is secondary or incidental genetic finding, and I would not like to chase it any further. Thank you very much, and we can open up now for discussion, and I'll stop sharing my screen.
Really marvelous for coming.
Rajiv, probably discussion we'll do at the end of the cases, because we are much ahead of time.
Yeah, perfect.
Okay. Problem, Rajiv, Dr. Shetty, I have only one problem. Each case scenario of this is different. Their genetic things are different. So do we want to discuss at the end, or should we discuss at each?
As you say, because as you say.
I think we should have a short discussion after every case, Rajiv, because this is different from a cardiomyopathy. Everything will be totally different. That is my problem.
I agree with you, Kane.
Okay.
So we will
what do you say, Kirkar? What do you say, Praful?
I agree. Let us do it case-based here.
Case-based. Yeah, let's complete this completely, because so Rajiv, please bear with us and let's finish, because this is different from something else which we are going to discuss. At the end, we will forget what Amit had discussed and what are the issues raised by Amit.
Ghansham, actually, Sadanand and me, we had one feeling that there were 5 case presentations, so should we keep it at the end? But I agree with Praful and you that because the cases are different, we can go ahead with each case discussion straight away as soon as the case is over. Just to begin the discussion, Amit, I just wanted to have I had 2 queries. One in your first case, because it was CPVT, we are not contemplating any anti-tachy pacing sequence or anything. So is it okay to put just a sub-Q ICD if you decide to go for ICD, because we are not contemplating any anti-tachy pacing in polymorphic VT, CPVT. And secondly, I had read somewhere that CPVT with electrical shocks, there can be electrical storm also, precipitation of electrical storm. So are we better off with just the left stellate ganglionectomy or this and beta blocker along with that? Or would you proceed with ICD as well?
Absolutely, Raju. So this is a practical situation that in CPVT, sudden death in the family, syncopal spells, repeated syncopal spells, despite being on beta blockers. So the sure short way, if we can save a life, it is by an ICD. We know that. But as you very rightly pointed out, that in children and in CPVT, ICD can become a serious nuisance. Because what happens is that if the child has a polymorphic VT, gets a shock, this shock will increase the sympathetic drive, and this sympathetic drive will cause another VTVF, another shock, and there have been incidences where there have been, a clear storm, VT storm happening. So VT storm is now being initiated and perpetuated by the ICD. So in fact, when the child was younger, the first time 4 years ago, did not even discuss about the ICD and just told about the stellate ganglionectomy. Now the child is grown. She's almost 19 years now. And in terms of giving an ICD versus stellate ganglionectomy, definitely becomes the choices in terms of therapy. But yes, I would say that stellate ganglionectomy precedes the ICD implantation in CPVT, because it has the safety margin of not only being very effective, there are good registry data available of sympathectomy working in patients with CPVT over a long term, and therefore we should offer stellate ganglionotomy as a first choice, and that's what I have done. Though I have discussed, everything with the family and also in terms of ICD, but I have pushed them that you go ahead with the stellate ganglionectomy. We see the clinical response with this, and over a period of time, if there is a need, we might have to consider an ICD, but no, it can be a challenge. And to answer your first question briefly, yes, one could easily consider a sub-Q ICD in a patient where you have pure VT, pure VS kind of a situation. But I think the sub-Q ICD has its own problems and challenges in terms of it being very expensive and again very limited overall trial data. And in the Indian context, I think we have done very few, mainly because of the cost issues.
And just another question.
Your second case, sorry, can I ask a question?
Yeah. Yes, please.
Okay, your second case, Amit, when you have done the Holter monitoring, the strip at the bottom shows a very tall peaked T wave. Now there is a condition in which you can get hypokalemic periodic paralysis as well as hyperkalemic periodic paralysis. We are looking only at the heart. Please look at other factors whether they are responsible for a syncope that she had. That's my only request.
Yeah, so whenever we look at Holter, I think we have to be very careful that Holter ECG over 24 hours, you see a lot of physiological variation. You should be able to differentiate what is physiological and what is pathological. And what is also important is correlate the Holter ECG change with the symptom of the patient. So this patient was completely asymptomatic all throughout. And in bradycardia, especially in children, and these are Holters, these are not ECGs. So when we have all these other ECG manifestations for various conditions, they are derived from a 12-lead ECG, proper ECG, and Holter and event monitors have a challenge in terms of the configuration of the morphology of the ECG. So you should do it with caution.
Dr. Kane, please.
Yeah, Amit, could you put up that genetic study slide? Because I wanted to ask the geneticists, let's know from them how would they interpret and what is their advice? Because you raised those queries. So let's sort out those queries with them because they are here and if they can help us. Like yeah.
So let me put up for the second case.
Yeah.
And the second case is, a family with Marfan. And now we have uncertain significance of MBN1 and uncertain significance of an RYR2 with probably CPVT. So now the geneticists can guide us.
Yeah, is Madam Shubha available with us or Dr.
Yes,
Snehal?
What is the phenotype of the patient? Why was the test done? Sorry, I missed some of the.
So as far as the phenotype is concerned, the child does have Marfan features. But no other symptoms. One syncopal spell, which is
so the Marfan features, the Marfanoid habitus doesn't make the diagnosis unless the GET criteria of cardiac involvement or ophthalmic involvement. So presence of Marfanoid features cannot be taken just as a phenotype for Marfan syndrome. So unless there is some cardiac evidence of aortic wall dilatation or specific findings for ophthalmological, then you can consider this as a phenotype. Secondly, if there are other individuals in the family who are affected, you can see if the affected individuals have the same variant. So that can add something to that. But as I said, if it is uncertain significance, then unless very characteristic phenotype is there for Marfan, you cannot use it as a causative variant.
Can I ask a question, Rajiv?
Yes, Akshay, and you have to go ahead inviting the second person also.
Yeah. Just one question. How does one decide that this RYR2 gene was found in this patient was of uncertain significance?
For that, the criteria of if this variant is not reported anywhere in the patient's database as well as normal population database, then it becomes a very, it is uncertain significance. So for that, there are some more criteria that if it is a nonsense variant, that is stop codon, or what is the other variants described in this patient. If there is same variant present in the codon or the same exon, then we can add this information to that. But otherwise, for a patient with Marfan syndrome phenotype, if you get this variant, then you can keep it not as a causative for Marfan syndrome, but because they are predisposed to malignant hyperthermia, you can inform them that probably it may be important as a pharmacogenetic variant and keep it in their record for if there is some surgery or anesthesia or something like that. And then the pathogenic variant for hemochromatosis. It's a heterozygous and it has a very variable course in the general population. That is why initially it was thought that for common pathogenic variants for hemochromatosis, general population screening can be done because the treatment is very easy, just blood donation. However, because the natural course for hemochromatosis is not well worked up, and hence I think I will not use this variant also. One can go ahead and do serum ferritin, etc. But these are non-invasive investigations. But for decision making, I will not use it for any further purposes. So for Marfan, again, you have to look for the definite features of Marfan, because we get many reference with just Marfanoid habitus and some evidence of some palpitation or tachycardia. That cannot be taken as a clinical diagnosis of. There are 10 criteria and they should be fulfilled.
Okay, shall we go to the next case?
Yes, yes.
Yeah. So we invite, there are two speakers for this next case, Dr. Phadke, Dr. Milind Phadke and Dr. Sriram Ishwaran. They will show a case of cardiomyopathy or maybe more cases of cardiomyopathy. Please go ahead.
So Dr. Sriram will be doing all the speaking. He is the one who is presenting. He's our third year registrar and he's presenting on such a senior forum. It's a good opportunity.
Absolutely, Milind. You as the backbone, he can present. Definitely, no doubt.
Very good morning to one and all. Am I audible?
Yeah. Yes, yes.
Is my screen visible right now?
Yes, yes, yes. Please go the full screen mode.
Yes, sir. A very good morning to one and all. Today I am going to be presenting a peculiar case of familial edema under the able guidance of Dr. Milind Phadke sir, who is the additional professor of cardiology at Sion Hospital. With special mention and thanks to Dr. Nitin Burkule sir, a consultant cardiologist at Jupiter Hospital, who has provided us with all the vital data with respect to diagnosing this particular case, and also with the able guidance of Dr. Pratap Nathani sir, the professor and head of cardiology at Sion Hospital, and Dr. Ajay Mahajan sir, the professor and head of cardiology at KM Hospital. So our patient is a 41-year-old male who basically presented with complaints of class 2 dyspnea and anasarca since the last 4 to 5 months. And he had these similar episodes in two maternal uncles, both of whom had succumbed to a similar mysterious illness. On examination, he was vitally stable, but he had significant anasarca. His JVP was elevated with decreased X waves and prominent Y descent, and there was CV complexes visible, and he had mild palpable tender hepatomegaly, and there was shifting evidence of free fluid in the abdomen. Routine investigations were unremarkable except for the fact that he had mild congestive hepatitis. He had a borderline creatinine of 1.5 milligram per deciliter and mild proteinuria, but he had significantly elevated cardiac troponins as well as NT-proBNP levels, which were correlating with the clinical diagnosis of heart failure. His ECG is as follows, which is suggestive of a normal sinus rhythm with a heart rate of 100 per minute, but he had significant Q waves in inferior as well as the anterior leads, and he had a very poor R wave progression. The 2D echography which we have done for this patient, it was suggestive of cross concentric hypertrophy of the left ventricle as well as right ventricular free wall thickening. Biatrial enlargement was seen, as well as there was thickening of the interatrial septa and the heart valves. We could also see that IVC was significantly congested and there was inspiratory diastolic flow reversal within the hepatic vein. These were all correlating with a suspicion of a restrictive cardiomyopathy-like features. His Doppler examination revealed that there was significant grade 3 diastolic dysfunction with an elevated E by A ratio of 3 and grossly reduced medial as well as lateral mitral annular tissue velocities. He had a severe tricuspid regurgitation with a TR jet velocity of 4 meters per second. GLS was done, which was suggestive of a globally reduced strain with apical sparing, with the characteristic stereontoc appearance, which was pointing towards cardiac amyloidosis. So with these features, on this suspicion, we went ahead with a cardiac MRI, which was suggestive of a transmural late gadolinium enhancement, which was seen in both the ventricles as well as thickened intraventricular septa. The heart valves were thickened, and there was these significant findings were correlating with cardiac amyloidosis. The next step was to rule out the presence of any free light chains or free globulins which were negative by immunofixation. And we did a technetium-99 pyrophosphate scintigraphy, which was positive for grade 3 uptake noted in the heart. So with these findings, we came with a definitive diagnosis of cardiac amyloidosis of the ATTR type, which does not require any tissue diagnosis. We did a genetic sequencing of this patient. As we see that he was positive for the pathogenic variant of the TTR variant, where there was a substitution of the aspartate with glutamate on the 38th position of the protein. And he was heterozygous for this mutation. Genetic testing of the mother revealed homozygosity for the same. So probably both his uncles also could have had the same disease and they succumbed to the same. He was initially responding very unreliably to the diuretics and routine heart failure management. Based on this diagnosis, we was advised oral tafamidis. We got this genetic testing from Dr. Ashwin Dalal sir, who helped us with genotyping from Hyderabad. But due to financial issues, the tafamidis tablet could not be procured. Over 1 year, he gradually worsened and he succumbed to heart failure. So let us basically deal with what this gamut of amyloidosis. Amyloidosis is a systemic disease which is characterized by extracellular deposition of misfolded proteins into beta pleated chains. It has a characteristic apple green birefringence which is seen under polarized microscopy, which is a pathognomonic sign. Tissue diagnosis is very essential for diagnosis until and unless ATTR type is definitely confirmed. The typical organs which are involved are peripheral nerves, the heart, the kidneys, GIT, and sometimes even liver and spleen. There are approximately around total 30 types of amyloid which affect the system, out of which only 9 types affect the heart, out of which 98% of them are caused by either AL or ATTR type. The ATTR has 2, genetic as well as wild type of cardiac amyloidosis. Other less variants have been mentioned. The systemic inflammatory amyloidosis, which is caused due to AA type, it is a very less common type. One of the other important caveats that we have to notice is that we have to do tissue typing of the type of amyloid, because that will help us in giving the right diagnosis, and so that appropriate therapy can be initiated for the patient. So this is just a summary of what are the clinical scenarios in which you suspect cardiac amyloidosis. With the left ventricular wall thickness of around more than 12 millimeters, in heart failure patients or with severe aortic stenosis, those patients who are more than 65 years of age, they have approximately 15% prevalence. So it is necessary that they are screened for the same. You have to look for any abnormal skin bruising, multiple peripheral neuropathies. There is proteinuria, the characteristic ECG findings of pseudo Q waves, AV conduction abnormalities, decreased QRS voltage, and in addition to that, any family history which is significant, these will all point towards cardiac amyloidosis.
Clarify one thing, Sriram, if you allow me.
Yes, sir.
Some of these extracardiac features that have been described, particularly these skeletal features like carpal tunnel syndrome, biceps tendon rupture, and lumbar canal stenosis, these are specific to the ATTR type of amyloidosis. They are not there in AL.
Sir. They are not there in AL.
Yeah, so what you get in AL is more of macroglossia, skin bruising, renal involvement. ATTR is very peculiar. It will involve only the heart and the nervous system. It doesn't involve the rest, while AL is more of a systemic disease.
Yes.
Okay, just go ahead.
So cardiac manifestations of amyloidosis are basically of the restrictive cardiomyopathy type predominantly, manifesting either as right or as left heart failure because of the tissue infiltration leading to reduced compliance. It can also be in the form of any conduction disturbances like atrial arrhythmias, atrial fibrillation, as well as they have because of the atrial fibrillation or even in sinus rhythm due to atrial dysfunction, they can develop thrombi, which can develop systemic thromboembolic manifestations. They can also have microvascular angina due to the small vessel infiltration. And orthostatic hypotension is quite a common feature because of autonomic dysfunction associated with cardiac amyloidosis. ECG findings are very characteristic, having a low QRS voltage to mass ratio, which is typically limb lead showing less than 5 mm, and it is seen hardly up to 40% of those patients. Pseudo infarct pattern, which is seen on the precordial leads, is seen in hardly half of the total population, and conduction abnormalities like atrial arrhythmias, complete heart block, or ventricular arrhythmias can also be common. 2D echo, which is suggestive of biventricular hypertrophy, thickening of the intraatrial septa, the heart valves, cross tissue Doppler abnormalities, the typical 555 sign, which is described for cardiac amyloidosis, where there is reduced tissue Doppler velocities of both the mitral annuli as well as the reduced TAPSE, which is seen, and also the reduced S- velocity for those patients, which is less than 5 centimeter per second, and the sparkling appearance of the myocardium, and grade 2 or more diastolic dysfunction, and congested IVC with restrictive features. The global longitudinal strain for these patients is quite pathognomonic. It is seen in 90% specificity of the patients with cardiac amyloidosis. It has the characteristic cherry on top appearance with basal involvement and apical sparing, as a result of which and it has a very good correlation with NT-proBNP levels as well as the cardiac troponin levels. It helps in also following up, though it is not a recommended way to follow, but it helps in following up these patients of cardiac amyloidosis significantly, along with the cardiac markers. So this is just a summary of cardiac MRI features which are suggestive of increased LGE, either subendocardial or transmural variant.
I'll just stop you at one point and just clarify that till now, whatever investigations he has said, they cannot differentiate between AL and ATTR. None of these investigations. ECG features, echo, everything is almost the same. What differentiates the two is the age of presentation, and for AL, documentation of a plasma cell dyscrasias. So that is now, once we know this is cardiac amyloid, that is now what we have to find out. Is this AL or AA? And ATTR or AL, and in ATTR, what are we dealing with? So that now he will go ahead. Even MRI cannot differentiate between the two.
Yes, sir. So first, in all of this, the first step is to rule out or rule in the presence of free light chains, either by serum free light chain ratio or by urine or serum immunofixation, which is the typical test which is used for diagnosing. And since AL or ATTR is the most common, we typically deal with only these two. So if in the presence of once we have ruled out the presence of free light chain, the next step I'll come back to the slide. The next step is logically to go for a technetium 99 scintigraphy, which has been described in 4 grades, where grade 0 is no cardiac uptake, grade 1 is uptake less than the skeletal uptake, grade 2 is uptake equal to skeletal uptake, and grade 3 where the cardiac uptake is much more than skeletal uptake. So out of these, the grade 2 and grade 3 uptake, even in the absence of tissue diagnosis, if there is a absence of free light chains, we can definitely, and with the clinical and echocardiographic features, we can give a 98% specific diagnosis of ATTR type of cardiac amyloidosis. Once we have diagnosed that, we can go ahead with the ATTR gene sequencing to rule out whether it is a wild type or it is a genetic variant. So as this small slide is present, it is just showing that there are it is divided into 2 scenarios, one which is most commonly causing cardiomyopathy, one which is most commonly causing neuropathy, and the ones which are causing mixed features. The most common ones which cause cardiomyopathy are the valine with 122 leucine substitution, which has a 100% cardiac involvement, and it is the most common one, but it typically affects a little later, more in the 6th decade of life, whereas the ones like valine 30 mutation with methionine, where the presentation can be earlier, but it has significant polyneuropathy as the manifestation than cardiac amyloidosis. This is just a summary of what I've already informed. So this is again how you approach a case of this thing. Once the tissue diagnosis in the form of endomyocardial biopsy is typically showing that apple green birefringence seen under polarized microscopy. Otherwise, there is overall you can see that this is just a summary of how to diagnose. The interpretation of this when you get a free light chain presence and in the presence of technetium pyrophosphate scintigraphy, if there is no uptake in both, the diagnosis is almost null and void. If there is hematological tests are negative, but you have a grade 1.
I'll just clarify this further. So the first step that you have to do when you're dealing with a diagnosed cardiac amyloidosis is that you have to do a light chain assay and you have to do urine and serum immune fixation. Mind you, routine electrophoresis and Benz-John protein is not enough. Strictly, all these three things have to be done. A light chain assay, a urine immunofixation, and a serum immunofixation, which is a little costly, but it's available routinely. Only if all three are negative and the technician scan has a grade 2 or 3 uptake, then you can say this is pakka ATTR, whether wild type or mutant. You don't require a tissue biopsy. But if you have one of the three tests positive and if you have an intermediate grade kind of technician scan, so grade 1 or grade 2 positive, then you have to go ahead with the myocardial biopsy. So you definitely first must rule out a plasma cell dyscrasias. All these three tests have to be negative. You have to show there is no monoclonal gammopathy. Then you can say that we don't need a tissue diagnosis. So that is exactly what the slide says. If you have a clear AL, fine. If you have a clear ATTR, fine. But in between, you need tissue diagnosis. Okay.
So the treatment is basically of two types. Either it can be of AL type of amyloidosis or ATTR type of diagnosis. Both of it vary significantly. AL type of amyloidosis is just the treatment of the main systemic disease of multiple myeloma or the cell plasma cell dyscrasia, which is with the melphalan based systemic chemotherapy. And a new approved one for systemic amyloidosis is daratumumab, which was approved in January 2021 by the US FDA, which is a monoclonal antibody against the plasma cells. But for ATTR diagnosis, it has three-step management. One is the suppression of the synthesis of the amyloid fibrils. The second one is to stabilize the fibrils, and third one is to help in clearing of these amyloid deposits. Till date, the most common one which was used was liver transplantation, since ATTR, the transthyretin protein, is significantly produced by the liver. So once liver transplantation was done, it used to reduce the synthesis. But nowadays, it is too harsh step which is taken. But now the only indication for liver transplantation is those who have significant polyneuropathy. They are with significant autonomic dysfunction. That is where liver transplantation comes into play to reduce further damage. Otherwise, we are the only US FDA approved treatment till date, which was approved in 2019, is tafamidis, which binds to the thyroxine binding part of the transthyretin, and hence it helps in clearing of those. It helps in stabilizing the transthyretin protein so that it doesn't get misfolded and get deposited. This has been found.
To clarify in one sentence, ATTR basically is a tetramer. It's a tetrameric protein. What happens in pathogenic amyloidosis is it dissociates into its dimers and monomers, and then these aggregate and they clump and form amyloid deposits. What tafamidis is that it stabilizes the normal tetramer. It prevents its dissociation into individual monomers and then its aggregation. So it is the one which has been found to have maximum benefit.
There are other trials which are ongoing, but none has yet received approval. And the treatment of complications, like if the patient is developing any conduction abnormalities like complete heart block or heart failure, the use of permanent pacemakers and guideline-based usage of CRTs can be done. And for thromboembolic manifestations, they need systemic anticoagulation. One important feature is that in cases of amyloidosis, the patients can have atrial thrombi even in sinus rhythm. Hence, these patients need to be screened for transesophageal presence of any atrial thrombi and be given systemic anticoagulation even though they are in sinus rhythm. These patients do not respond to the routine heart failure managements like diuretics and beta blockers and ACE inhibitors because of the coexisting autonomic neuropathy, and hence they may behave differently. And finally, those patients who have aortic stenosis, they have a very good outcome with transcatheter aortic valve replacement than open valve replacement. And other than that, it is just routine management of the coexisting complications. And lastly, but not the least, the final step could be heart transplant. But until and unless the primary disease is managed and controlled significantly, even heart transplant may not be definitively credible for the treatment. Overall, the prognosis of these patients, it is not, still long-term studies are undergoing because hardly around 3 to 4 years of studies are available, and they all suggest a good this for tafamidis. But in case of AL amyloidosis, if they undergo hematopoietic stem cell transplantation, it has been found that some studies show that even survival up to 10 to 15 years have been noted. But the overall prognosis is still a little in the gray zone only. And what we have to monitor is routine yearly 2D ecographies, and we have to do cardiac MRIs once a year to look at whether it is progressing or it is worsening. And with routine monitoring of the markers like NT-proBNP levels and cardiac troponin levels. And in case of AL amyloidosis, the level of the serum free light chains, which are used for routine monitoring. And in case of ATTR amyloidosis, the effective glomerular filtration or estimated glomerular filtration rate with to notice the creatinine clearance is one more method. In addition to that, they need to be monitored.
Summarize a little bit. We are a little short of time. So.
Hello, can I ask a question, please?
Yes,
I wanted to find out about the family members, where the parents tested for the sequence variation. What are their ECG echo and other findings, whether they are suggestive of any disease process or not, and thirdly, whether this gene is completely penetrant or not. That means everybody who has the mutation will have the disease or not. Because if parents are asymptomatic and they have the same, one of them has the same variant, how to interpret? So these three information are necessary before making a decision about family members.
And I would like to add few things, because this is an excellent case where you are approaching.
Can we go to the gallery view, please? If he has finished the presentation, then it might be better to discuss that way.
This is the last slide. This is the last slide.
That's an excellent presentation. Yeah.
Yes.
So coming to the genetics of it, because this was familial, therefore the genetics comes into play, isn't it? Otherwise, it was as a case report. And from treatment point of view, I just want to, before we get into the genetics, tafamidis is available on compassionate grounds. In fact, we are maintaining a registry at the KEM about such patients, and we get those. We have got a good liaison with Tatas who do a bone scan, which is very simple to interpret. And that is the basis of all this. Just doing we pick up on imaging very fast. And you've showed it so well with your GLS and cherry on top, etc. After that, as Dr. Milind Phadke pointed out, the hematological screen, and then directly you can, basis the bone scan, come to a diagnosis, isn't it? And then to maintain a registry becomes important, because although tafamidis is not commercially yet available on compassionate grounds, it's available through Pfizer. And in fact, with Pfizer, we are countrywide coming out with a document from India's perspective also on diagnosis of amyloid. Now, coming to the genetics of it, had this not been the family history out here, there was not a need in this patient, if there was no family history, to really get into the genetics.
Excuse me, sir. I slightly differ from that. Yes. Because there may be family members affected or unaffected. Many autosomal recessive and dominant disorders usually do not have family history, and the first individual who is presenting may be the. So
absolutely, madam. Had this patient not had a family history,
and even now at
we have done genetics
because the relatives are related through mother, and mother whether she is carrying the same sequence variation or not, that I didn't realize that whether
a mother has the same.
I want to have few points.
May I clarify about this case?
Few points before you clarify, Dr. Milind, that we have this familial amyloidosis case. I have two families now. As Madam rightly put forth, that you have a case of cardiomyopathy, you have a polyneuropathy, you may have somebody in the family with liver disease, unknown liver disease, sudden cardiac death. This is perfectly fitting into familial amyloidosis phenotype. So in some patients, even before you take up a biopsy, you can directly go ahead for genetic testing, even if you have a few members with few symptomatology and one member conforming to a phenotype of familial amyloidosis. So biopsy can be averted and directly genetic testing can be offered. Family members need to screen. Now, patients in amyloidosis come to a cardiologist maybe when cardiomyopathy sets in. I have a family where two members are having liver manifestation, one is having polyneuropathy and one is completely with cardiomyopathy. Now these liver patients or these neuropathy patients can definitely be referred for liver transplantation and cardiac transplantation. So even if a family member is coming to you for a cardiomyopathy, if you have other family members which can be helped for liver transplantation, because at Apollo hospitals now we are collecting these patients where we are screening family members, can be referred for liver transplantation, can be cured before cardiomyopathy. Before once that cardiomyopathy sets in, the transplant team is the patient, they are very not keen in taking for transplants. So that is how the genetics may help. And there are few antisense oligonucleotide already in phase 3 trials for amyloidosis patients, which may come in soon. These drugs can definitely help in cure. So as Madam said, third generation pedigree, clinical examination, even if one case of cardiomyopathy, whether any other child born with dysmorphic features may point to syndromes like Noonan, when other family members, some of them may have polyneuropathy, which may fits into. So one cardiomyopathy may diverge into so many conditions if there is a complete family history, and then you fit into syndromic or non-syndromic, and you may take up nerve biopsies in some of the family members' polyneuropathy. I don't know how feasible or how endocardial biopsy every patient you will be able to take, but nerve biopsy, which is a little less invasive, is also helpful.
So the point is this, if I can clarify, this case came to us about 2 years ago. We were still also not too much aware about amyloidosis at that time. So the reason for testing this patient was, the first thing was the age. So the age was around 45 years, and amyloidosis, senile type of amyloidosis or ATTR wild type, it is typically a disease after 60 or 65. So there were only two possibilities in this patient. Once we got the peculiar cherry on top appearance and a restrictive cardiomyopathy, this was likely amyloidosis. So we only had to clarify whether this was AL, because the treatment of AL is totally different. You have to treat the plasma cell dyscrasias. And for the mutation, it was important to screen the family. So we have screened the mother, and the mother has a similar mutation, as Dr. Sriram pointed out. Unfortunately, the patient comes from outside Mumbai. It is not easy to get the family here, and especially since the patient has passed away, they are not too keen on coming. But yes, I would definitely screen the entire family with an echo, including a strain imaging also, and would try my best to get gene sequence.
So that is why I stressed on DNA storage. If a patient coming to you at a very last stage, not getting investigation, you can still store the DNA, and which will help in family members. And second point I was stressing was, if this sudden, the family member screening to get a complete family history. And third point may be to detect members where we can help by transplantation. So that is how genetic diagnosis. And first and fourth point, that sampling is very easy now, can be picked up even if patient is in the remote area, the relative is in the remote area. We can easily pick up the sample, take out the DNA, store. So it is definitely not a hurdle now anymore.
It is a hurdle to get the patients to be willing for all this. The cost,
of course.
It's not so easy. There is a lot
I understand that all cardiomyopathies, this patient is, of course, because he presented young amyloid, therefore to do up some workup. Do I understand that all cardiomyopathies therefore should get genetic screening? We go by clinical cascading first, isn't it? You want to do a family tree, all that you said, 3 generations, etc. After that, do we go for genetics, or do we go for genetics upfront? Then we have so many VUSs out there. I'm asking you the clinical indication for doing a genetic screening without a family history. In this case, there is no question. So if you are telling me that all amyloids should get a genetic screen, I do not know really. What is the implication of these diagnoses to family members?
No. We are not saying all amyloid should get a genetic screening. The red flag in this was the age. And the fact
exactly. I totally agree.
Amyloid. The question is, should all cardiomyopathies get a genetic?
Exactly. That is what I'm asking.
All dilated cardiomyopathies. That is my question. Yeah. Should all DCMs get a genetic screen?
Yes. I would say that all cardiomyopathies and all phenotypes which have overlapping phenotypes with monogenic disorders should get genetic testing, exome sequencing. And it is not screening, but it is really testing.
Unfortunately, our clinical guidelines don't say that. And like what Raju Karnik pointed out, it's at the 7th level, the investigation of genetic testing. In this case, I totally agree that genetic screen is necessary. I'm talking about in general for all cardiomyopathies. We have dilated hypertrophic cardiomyopathies, as well as now we have got especially the amyloid cardiomyopathy. In the workup of an amyloid cardiomyopathy, as what Dr. Phadke and Dr. Ishwaran pointed out, the genetic screen comes only in certain specific instances. It does not come for all.
Yes.
I'm just making that point.
I mean, I would just like to add, because like Madam has pointed out and everybody is pointing out, we do not have Indian data how much cardiomyopathy % of patients we should immediately apply. So what I had written in a slide, that estimation of cardiac genetic clinics. So if we comprehensively work together, maybe we, for our Indian scenarios, that which type of patients accept and don't
accept. I
agree
with that. I think we have to come out with our own data, and it's good to do in most of the patients so that we learn from it. I would say
it will probably be the first reported case
and
of
Burkule, we would love to have some opinion from Dr. Burkule, Dr. Akshay, and please take over.
One question about cardiomyopathy, Dr. Snehal. Till now, does genetic testing help in prognosticating the hypertrophic cardiomyopathy patients as far as sudden death is concerned? For sudden death, has it been useful?
Yes, sir. I would like to quote the case which was excellently discussed by Dr. Amit, and which I had also put of channelopathy, and I had put one case of dilated cardiomyopathy. And it will also cover Dr. Kerker's question that it has become nowadays very personalized, that whether I should apply genetic testing in that particular family. Like you have a case of cardiomyopathy and you have multiple members affected, you may suspect amyloidosis. So what will be application of genetic testing in that particular family? Whether you are going to prevent any death, like there is only a single child and you are not going to do in siblings, then you can just say that whether it will help in mother or father or other first degree relatives. So I think that discussion would be henceforth very personalized, that in that particular family, whether you have a risk of sudden cardiac death, whether you have associated atherosclerosis CAD at risk, or whether you have any other family history contributing, or investigations for that matter contributing, which will help you in preventing the management making in the family members, and then you discuss genetic testing with the family. So that helps in that will actually help taking out cases which where you immediately advise genetic testing, where genetic testing can be put up as a follow-up test when patient comes to you as a follow-up, or when, like he said, that after cardiac MRI or after doing ECGs on multiple times. So we have to categorize now patients or families like this.
Dr. Akshay, specifically as a clinician, to talk about hypertrophic cardiomyopathy, there are certain mutations in which the presentation is earlier and the first presentation can be sudden cardiac death. So it's very important to know which mutation we are dealing with. And in these patients, the familial screening has to start very early, maybe in even childhood or adolescence. So having that mutation, in fact, some of the risk scores are now incorporating this mutational analysis also. So there are certain high risk mutations, it adds 1 or 2 points to your risk scores and your need for an ICD. So in HCM especially, it is very, very important.
All right. Okay.
If I may interrupt, especially related to this case, in Circulation Journal, it was mentioned that even those patients who present with familial cardiac amyloidosis, it is not a routine advice to screen all of them, because all of them have adult presentation. Preferably, once they have attained like more than 18 or 20 years, it is better to screen. And what they have said is that in these families, the age of presentation of the patient is first noted, and the family started to be screened at least 10 years prior to the age of presentation. So like in this case, since he presented at around 40-41 years of age, the ideal time to start his familial screening for his close relatives would be from the age of 30 years and not the pan-family screening.
Okay, now can I just intervene? I think, Rajiv, we can move to the next, because I know the discussion is interesting. Yes. We can be here till evening, but let's give enough chance to because we have 3 more cases to be presented.
Yes, Dr. Kane. Yeah. Dr. Kane, unless you announce that there is no time limit for this entire
there is no time limit unless we are tired. There is no time limit.
So,
I
all
confirmed
right.
it with Vama people. They've been gracious to say you can extend the timing. So long as we are all
maybe
keen
one
on
comment
learning
from
and
Burkale
discussing,
sir,
we have
because
no problem.
Burkale sir has a lot of experience in this and he helped us work up this case.
Yeah. I think he also has a case of cardiomyopathy to present. So maybe that time he can cover up this also, Burkale. Keep it for that. And I would now go to the next, whatever. I don't know who is the next in line.
Yes, Dr. Kane, let us go to the next talk, which is on dyslipidemia, hyperlipidemia in the first decade. I remember, in fact, maybe 15-20 years ago, when Enas was in India, I had a personal discussion with him, and he was saying that he's a president of coronary artery disease in India, CADI Society. So he was saying that in USA, in fact, in Asian population, he does in the the school health clinic only, he prefers to do the lipid panel. And if it is abnormal, then diet, exercise, lifestyle modification. And by the age of 10 years, still if the child is with dyslipidemia, he wouldn't mind going for, at that time, of course, 20 years ago, it was only statin, not ezetimibe, not PCSK9. But with this PCSK9 and then Mendelian randomization studies and loss of mutation, loss of function studies, we have very good data. And then I'm sure this Dr. Praveen Kulkarni will enlighten us on these dyslipidemias in young children, especially familial hyperlipidemias, and then our current modalities of treatment with genetics in focus. Dr. Praveen, please.
Thanks, Dr. Karnik, and it's been a wonderful session so far, very enlightening, as all of us must have realized. And also reviewing this particular topic that I am going to present now has actually brought in a new understanding, a new perspective that we need to look into, even in general as adult, largely managing adult patients, when we need to have a perspective in terms of a genetic predisposition. The topic that I'll be covering today is a dyslipidemia in children, and largely it is driven by a fact that I had a fairly sad but interesting case. This is the index case of a 4-year-old girl who comes with actually very limited resources. She saw an endocrinologist for the skin lesions that were rapidly growing. They presented as painless yellowish non-tender papules and nodules, largely beginning around the knees, the elbows, and then progressing on to involve the buttocks as well. So straight away, as suspected, there were cholesterol-laden skin lesions. Labs were ordered, and not surprisingly, we saw gross dyslipidemia, but the extent of dyslipidemia is what took us by surprise. The total cholesterol was over 700, largely driven by a rise in the LDL cholesterol of over 680. The triglycerides being normal in her case. So mind you, she was only 4. By the time she came to me, she was 5. There is no obvious family history forthcoming to suggest a premature coronary artery disease. She's the only child. Though the extended family was not screened, we screened her parents. The mother did not have any gross dyslipidemia. The father has dyslipidemia with a total cholesterol of 298 and the LDL of 170. So in the setting of severe and gross hypercholesterolemia with typical skin lesions, a possible diagnosis, a clinical possible diagnosis of familial homozygous hypercholesterolemia was made. She was 6 when she came to me. On reviewing the literature, there isn't much to initiate treatment at such a tender age. But there have been exceptions where simvastatin and atorvastatin has been tried, particularly in familial homozygous hypercholesterolemia. And we started her on very low doses of simvastatin, titrating went up to 40 mg. But the lipid profile just did not budge. Even today, she remains on 40 mg, but it has not at all moved an inch. The LDL cholesterol remains well over 600. So now we move on to childhood dyslipidemia. So so basically it would be primary and secondary. Secondary, we're all very well aware. Secondary would be nephrotic syndrome, diabetes, obesity. what we are primarily interested is to see whether there is an inherited disorder which would now be qualified under the primary dyslipidemic syndromes. So they could be either single or multiple gene mutations, and they're often responsible for very severe form of lipid abnormalities. Now, an interesting study that I came up is if someone has a proven genetic mutation, particularly for a familial hypercholesterolemia mutation, a similar level of LDL cholesterol, there compared to mutation negative hypercholesterolemia, there's a 22% higher risk of developing premature coronary artery disease. So again, reinforcing the need to assess genetic for genetic mutations in these patients, where we can predict that they're at a much higher risk for developing eventual coronary artery disease. So a quick look on secondary dyslipidemia. So we know the common causes which we need to screen for. There's a big and exhaustive list of secondary other causes causing secondary dyslipidemia, but thyroid disorders, nephrotic syndrome, and liver disease are the prominent ones that we need to routinely screen for. Now, lipid metabolism, when I was reviewing this topic, I thought we'll just put in a single slide just for a quick recap. I'm sure most of us are very well aware about how lipid metabolism and how dyslipidemia is based on this lipid metabolism. So an exogenous lipoprotein metabolism, there's endogenous lipoprotein metabolism, and there's reverse cholesterol transport. Abnormality in any of these levels, for chylomicrons through lipoprotein lipase became chylomicron remnants, taken up by liver, becomes VLDL, IDL, and LDL, and taken back into the liver. And then there's a reverse cholesterol, which is driven by HDL primarily. So abnormality in any of these pathways can lead to dyslipidemia. So there are age-adjusted values, normative data given by multiple guidelines and multiple societies. This is one of the representative charts which has come up in the NHLBI statement, where the values for individual parameters within the lipid profile has been clearly stated. So the traditional classification of dyslipidemia was the Fredrickson's phenotype, and it dates back to 1967. Where it is classified into 1, 2, 2A, 2B, 3, 4, and 5. Again, if we go back to this lipid metabolism, we realize that aberrations in the pathway for this lipid metabolism is what finally correlates with this classification. What is more relevant now, and coming to the genetic predisposition causing dyslipidemia, what is more relevant now is to have a genetic correlation with the traditional dyslipidemia. So if I can spend a few minutes on this slide, it's a busy looking slide, but I'll just go through the types and the genetic disease that it correlates with. A type 1 disease where there is more of hyperchylomicrons is primarily driven by lipoprotein lipase deficiency. So chylomicrons, lipoprotein lipase deficiency will cause hyperchylomicronemia. Then type 2 is predominantly where there is hypercholesterolemia, where predominantly there is high LDL levels. And if you see the genetic defect, it can be monogenic or polygenic. There is an LDL receptor abnormality predominantly, which gives rise to familial hypercholesterolemia. There is also a familial combined hyperlipidemia where there is LDL and triglyceride rise. Also quickly naming 3, 4, and 5, where there is familial dys-beta lipoproteinemia, familial hypertriglyceridemia, and hyper-beta lipoproteinemia. So these are not very common in our clinical practice. So what is more relevant for us is moving from the Fredrickson's classification to genetic disorders causing lipoprotein metabolism. In this, the commonest one and the ones that we would frequently be facing is the heterozygous familial hypercholesterolemia, homozygous familial hypercholesterolemia, familial combined hypercholesterolemia, and then there are various others which I previously mentioned. So we'll spend a few slides moving through the familial hypercholesterolemia. Amongst the commonest one is the familial combined hyperlipidemia. It's highly prevalent with a prevalence of close to 1 in 100 to 1 in 200, where there is an elevation of both LDL cholesterol and triglycerides, and it's an increasing, an important cause of premature coronary artery disease. This, however, is likely polygenic. So there's no single gene that has been associated or a mutation that has been associated causing this. Now, this mixed lipidemia is known to predispose the patient for premature CAD. Again, considering family history, again, is very relevant once you know that an individual has a mixed dyslipidemia, to screen for the family as well, and also look for a family history of premature heart disease. The most important topic, and probably what relates to our index case, is also a familial hypercholesterolemia. Now, it's a monogenic disease caused by loss of function mutation in the LDL receptor or the ApoP genes, or it's a gain of function mutation in the PCSK9 gene. Majority of the cases are, however, caused by the LDLR receptor gene mutation, wherein the LDL uptake from the liver cells is impaired, eventually causing gross elevations in LDL cholesterol. There are 2 or 3 frequently used criteria to confirm the presence of familial hypercholesterolemia. The Dutch lipid network diagnostic criteria, and if we go back to this case, it is defined, the pointers that we take to confirm this FH is through family history, clinical history, physical examination, LDL levels, and DNA analysis. Now, in this case, we have not been able to do a DNA analysis because of the financial constraints. I'm, however, trying to get in touch with the societies, and we'll eventually get the genetic assessment done for this child. If you look into this case, this girl clearly has, though not tendinous, she has more of tuberous xanthomas and an LDL cholesterol which is grossly elevated. So purely as a single point marker, if you have levels of more than 300, the score goes up to 8, where it eventually it fits into a probable familial hypercholesterolemia diagnosis. And in the presence of typical xanthometas, the diagnosis is almost always confirmed. Though, of course, DNA analysis has a great role, genetic analysis has a great role here. If we move on to a Simon-Broom diagnostic criteria, we'll not go into the finer points here. If you see point 3 is a DNA-based assessment, the presence of just that, a positive study just based on a genetic assessment is enough to confirm familial hypercholesterolemia. Also, clinical parameters can also guide us into diagnosing FC in these cases. So the more common variant of the familial hypercholesterolemia is the heterogeneous variety. It's a common codominant monogenic dyslipidemia, and it's quite common to the tune of 1 in 200 to 250. Homozygous, however, is uncommon. The prevalence rate is said to be around 1 in 1.6 lakhs to 1 in a million. They predominantly present with extensive xanthomas, marked premature and progressive cardiovascular disease. Total cholesterol typically is in the range of more than 500, as in this case. Most patients untreated develop coronary artery disease, aortic stenosis in the second decade or third decade of life, and typical survival is said to be around 30 years. I'll not spend much time
I mean,
here.
I may interrupt you. I think you'll have to cut short and go, because we have 2 more presentations and we are already 40 minutes beyond time.
Yes,
so
yeah.
bear with us. I want the presentations to be done. We are not going to allow any discussion on at least now, because then otherwise we won't be having time. But please complete as fast as.
No, there's a couple, just 3 or 4 slides. So familial beta dyslipidemia, and there is also familial hypertriglyceridemia. So why do we screen for dyslipidemia? So, you know, adult levels of lipid levels are almost achieved by 2 years, though there are variations during adolescence, puberty, and finally adulthood. If there is a strong family history of premature heart disease, gross dyslipidemia, studies, the guidelines suggest that the kids should be screened at as young an age as 2 years itself, and then 3 to 5 years annually till they reach adulthood. Various societies have come up with their own measures to screen these kids. The treatment is now standardized. I'll not spend much time here. Statins, however, we must realize have been studied typically from the age group of 8 and 10 and above, and not at the in the first decade of life. Increasingly, we have had now reports of, again in 8 years and above age group, where the PCSK9 inhibitors have also been used and have been found to be safe. The other two treatment options remain apheresis, which is, however, a 2-weekly treatment, and finally liver transplant when there is an LDLR receptor-related homozygous familial hypercholesterolemia, where it is a cure in itself. There have been multiple reports of liver transplant in these cases. Yeah. So I think I'll end there here. So we should all be aware of these monogenic and polygenic primary pediatric dyslipidemia, and a screen for them increasingly, which will help us risk assess and manage our patients better.
Yeah. Thank you, Praveen and Rajiv. I'll interrupt. Let us go straight to Dr. Prashant Bobate's presentation. And I would request Prashant, as a special request, try to make it short, and so that we can also go to Dr. Burkule's presentation, and we can have discussion of all these three at one shot at the end of the presentation.
Sorry, good afternoon.
So just highlight conotruncal genetic concerns, Prashant. Bear with me.
Yes, I will just, my talk is, I mean, the case is just concerning the genetic concerns of conotruncal abnormalities. Okay. So, if possible, I would like comments from the geneticists who are there on the...
but we are going to have it at the end of the two cases. Yes.
Yeah. So we have a 22-year-old female who was from a remote district, remote place from Ratnagiri. She was third in birth order, born to a third-degree consanguineous marriage, was diagnosed to have congenital heart disease at 3 years of life, but not operated for social reasons. She had easy fatigue, was in functional class 2, had cyanosis at rest with saturation of around 85%, which was increasing on exertion, had a history of recurrent admissions for respiratory tract infections when she was a child, but not so much now. She was good in Marathi literature. I was very poor in math, and there was no family history of any congenital heart disease. She was a short patient, around 146 centimeters, weighing around 36 kilograms, with saturation of 83%, mild clubbing. Nothing remarkable in the cardiovascular examination apart from an ejection systolic murmur and a vascular click. She didn't allow me to take a photograph. However, this is a representative photograph from the internet. We can see she had a flat faces, had micrognathia, and if you can see the nose, it was quite pathognomic of a disease which is called as a DiGeorge syndrome, which Dr. Snail had described before. This was an ECG, was similar, was nothing unremarkable, a routine ECG for a tetralogy of fellow. This is an X-ray, her X-ray, which is showing a right aortic arch. So an echocardiogram again, large subaortic VSD with severe pulmonary stenosis. Cardiac catheterization, also showing same severe hypoplastic pulmonary valve, confluent good sized branch pulmonary arteries, large subaortic VSD, a right aortic arch, no significant collaterals. So my question here would have been whether a genetic testing is warranted at this stage, and if yes, what would you would like to recommend and why? I would go directly. So we did a FISH test in this patient, and the 22q11 deletion was found to be positive. And why should we do the FISH test prior to the surgery? I'll discuss a little bit, 1 or 2 slides. So most of the conotruncal abnormalities are associated with DiGeorge syndrome, especially interrupted aortic arch, truncus arteriosus. Tetralogy of Fallot, if it is associated with aortic arch abnormalities, especially right aortic arch or right aortic arch with an aberrant subclavian, the incidence of DiGeorge syndrome is quite high, ranging from around 15 to almost 40%. Why should we do it? There are other non-cardiac manifestations of DiGeorge syndrome. Facial abnormalities can be seen in almost 100%. Speech and learning difficulties can be seen. In smaller patients, will have neonatal hypocalcemia, T-cell deficiency, skeletal abnormalities, and renal malformations. This was a FISH test which was positive. Pre-op assessment is important because especially in small children, not so much with her, but if you have a 6-month-old child of tetralogy of Fallot, it is very, very important to do this testing because they are immunodeficient. Prompt and aggressive treatment of infections and irradiated CME negative blood has to be used. Thrombocytopenia, hypocalcemia, velopharyngeal abnormalities have to be ruled out. Vasomotor instability has to be treated with aggressive ionotropes. Upper cervical spine and craniovertebral abnormalities are there, which could be concerns during positioning during anesthesia, and also have nephro and neuro issues which need to be ruled out. And one thing which I forgot to mention here is sometimes they have significant gastroesophageal reflux, especially in the infancy, and those need to be addressed during and after surgery in the post-operative period. Our patient underwent an intracardiac repair with a biprosthetic valve in 2018, had an uneventful post-operative course, was on regular follow-up on echosprin. However, last year she came back to us and she wanted now to get married. And the question is, how do we approach this now? What is the role of prenatal and pre-pregnancy counseling? Who is the right person to do the same? What is the risk of the fetus getting affected? And would it be possible to diagnose and prognosticate DiGeorge syndrome antenatally? I had one more slide after this. With all of these was done, genetic counseling was done, and she was told to have antenatal screening. However, she didn't follow up. She directly came to me in the 5th month of gestation, a month back, and we can see the child has got a tetralogy of Fallot. There is overriding of aorta, and has an absent pulmonary valve syndrome. Thank you. I would really appreciate inputs from the genetics on this, maybe after the next case is discussed.
Yeah. So we'll go straight to Dr. Burkule, and at the end of Burkule's talk, we'll discuss all the 3 cases. Dr. Burkule, please share your screen. Thanks, Prashant, for a very excellent presentation. I'm sure we have our faculty with us, so we can go for that discussion.
Thank you. Thank you, sir.
Burkule sir, give us the vital points so that
is my screen visible?
Yes, you are audible and your screen is visible very much. Please go ahead.
Yeah. So this is very quickly I'll go through it. A 22-year-old male, shortness of breath on exertion. He had difficulty in climbing staircases and riding bicycle. No history of rheumatic heart disease, blood pressure, diabetes. So he's referred for an echo evaluation for his shortness of breath. This is his ECG, which is not does not have any remarkable signs. This is echo, and you can see that the heart is dilated and the posterior wall looks akinetic there. The LV size is enlarged. This is the 4-chamber and 2-chamber, showing that the lateral wall and the inferior wall looks significantly hypokinetic. And this is a short axis view, and one may feel as if the posterior lateral wall, is there an infarct or something? It looks really thinned out and almost akinetic. The EF is low, and the E by A is about 2. The E primes are low, and the E by E prime is around 9, not very high. The global longitudinal strain doesn't show any specific pattern, though it is reduced to 15%. And the strains do not show any dyssynchrony as such. The walls show quite a good synchrony. But if you look at the left atrial volume, which is perfectly normal, but interestingly, if you see the LV rotation, the base should always rotate clockwise, but it is moving anti-clockwise. The apex should always rotate anti-clockwise. It is rotating anti-clockwise. So both the base and apex is rotating anti-clockwise. And this is called as solid body rotation. So that tells you that the spiral myocardial fibers are affected. The left atrial strain is significantly reduced. And this is the patient's cardiac MRI. The MRI also shows a dilated ventricle, and you can see the posterior wall looks akinetic. And the ejection fraction is around 40%. And even the short axis stack show you that the posterolateral wall is quite thin and akinetic. And this is the feature tracking strain on the MRI, which is around 14%, which is almost close to the echo speckle tracking longitudinal strain. And this is the patient's late gadolinium enhancement. And what you see interestingly is that if you look at the lateral and posterior and almost inferior wall, there is a mid to epicardial enhancement. And this is particularly very characteristic of the involvement in very few diseases, one of which is the skeletal muscular dystrophies, which this particular patient has. And in other cases, it is seen in Fabry's, but of course, that will be also associated with myocardial thickness. That means it will be more of a mimicker of an HCM rather than a DCM. So this is very classical in the muscular dystrophies. And this is putting the slides together, and you can see the areas which are akinetic. You also have the epicardial to mid myocardial late gadolinium enhancement. So it is perfectly corresponding with the function. So the thing is, these are the maps which show which is the T1 maps are higher, and this is the patient's lower limb examination, and you can see the pseudo hypertrophy of the calf muscles. So these are the different muscular dystrophies, and these are the distributions. And what we are dealing with here is the Becker's, and this has got a distribution of this is the particular the distribution for both the calves and the thighs and also the shoulder. This is the Emery-Dreyfus. This is the only limb girdle. This is the facioscapular and humeral. And this is only distal, and this is oculopharyngeal. Now, what is the importance of this is because now these are the skeletal muscles. This is the sarcolemma, and this is the extracellular matrix. Now, this sarcolemma should be fixed to the extracellular matrix, and this is done by a large glycoprotein complex. And these are the various proteins. If they have an abnormality, they lead to both skeletal and cardiac muscle myopathies. So of which one of the protein is dystrophin, but you also have this the dystroglycans. You have got also these sarcoglycan complex, integrin, then caveolin, so on and so forth. So all this entire glycoprotein complex is very important to anchor the sarcolemma to the extracellular matrix. And dystrophin has a role to prevent this glycoprotein complex getting destructed by the proteases. So when it is absent, then this becomes completely unstable. But each one of this member protein has got a genetic coding, and that when it is absent, so these are the genes, and these are the proteins encoded by these genes or this particular complex which anchors the sarcolemma. So what we are dealing here is the dystrophin, and the DMD is a gene. And generally the manifestations are Duchenne's or Becker muscular dystrophy. And along with that, you get a cardiomyopathy. So also for the sarcoglycans and also for the dystrobrevin and caveolin, you have got limb girdle myopathies, limb girdle muscular dystrophies. So it is very important that our neurologist also send us the patients with muscular dystrophy. Well, this was sent by neurologist itself. So muscular dystrophies for the echocardiographic screening, and then they should have the particular gene testings. So the dystrophinopathies are X-linked recessive. This is their incidence. And when it is presented very early, it is called Duchenne's. When it is presents late, it is called Becker's muscular dystrophy. And generally, you will have the Duchenne's associated dilated cardiomyopathy. Now, it produces extensive fibrosis of posterolateral LV wall, mitral regurgitation. Generally, the DMD, 1/3 of the patient by age of 14 will show the cardiomyopathy, while it is variable in presence of the Becker's muscular dystrophy. The diagnosis is showing the total CPK 5 times more than the upper limit of normal. Getting the dystrophin gene in genetic testing. If you do a muscle biopsy, then in Duchenne's, there is absent dystrophin on immunohistochemistry. Or in Becker's muscular dystrophy, it is sparse. It is not completely absent. But very interesting thing is to do a simple total CPK and the dystrophin gene analysis and this particular echo and MRI features. The treatment is very interesting because the glucocorticoids are shown to improve a lot of attributes of this muscular dystrophies. But from the cardiac point of view, surveillance is very important with ECG and echo and Holter. And so also the generally as they grow, the echo window becomes poor and you have to resort to cardiac MRI. And for the female carriers who are heterozygous also, you have to screen them every 3 to 5 years for the cardiomyopathy. And more important from the cardiologist perspective is that in these patients, starting ACE inhibitors and mineralocorticoid receptor antagonists actually have shown to delay the progress of this cardiomyopathy. So definitely surveillance is important because it has got direct clinical importance to start the neurohumoral blocking drugs as early as possible, especially the perindopril and carvedilol, so that you can postpone the eventual development of heart failure. We'll forget about the emeritus. But this was the question that Dr. Kerkar wanted to ask, whether does all cardiomyopathy need this kind of genetic sequencing? Actually, the American Heart Association guidelines say that first is that when you have a cardiomyopathy, rule out the secondary causes. That means if you have got HCM, you will rule out the HCM mimickers like Fabry's, amyloid, and so on. If you have dilated cardiomyopathy, you will rule out other causes of inflammatory cardiomyopathies or sarcoid or so on and so forth. Then you do a 3-generation pedigree chart. And in that, if you have 2 family members who have a history of a cardiomyopathy or a heart failure or a need of a pacemaker or an ICD, or if one of the family member had a sudden cardiac death, then think about doing the genetic testing. Thank you. I think Dr. Kane, you are muted.
What is happening? Okay. Yeah, no, your cases, all these 3 presentations could be a conference by itself. I realize that. So I think I have pushed too many things into a short time. So now it's open for discussion. Rajiv, are you there? Akshay and Snehal, and I think Madam has left. So Snehal, Prashant.
Dr. Rajiv also had another meeting, so maybe he's left, I think.
Okay, so Rajiv is there. Rajiv Karnik is there. So Prashant Bobati had a question. If you can answer that. Prashant, can you come back? Prashant, are you there?
Yes, sir. Yes, I'm there.
Yeah. What was your?
I'll just share my screen.
Yes. Yes.
Yeah, if you can. Yeah. Now we have enough time. So long as we are all interested, we are there.
No, so I'll just summarize. Yeah, first, very excellent cases. I've been all cardiologists as excellent managing cardiology. Also, I've started managing genetics. So I don't know whether I need to have, but for particularly for Dr. Prashant's case, and because the mother is now 19 weeks, I would stress again the importance of pre-test and post-test counseling, because this mother had to be stressed that how severe the disease can be in the fetus. There is variable presentation. She must have assumed that because she has got operated at adulthood and now she is well, her fetus may not be severely affected. So that is important before pregnancy, and prenatal diagnosis by Korean villus biopsy at 12 to 14 weeks, by amniocentesis at 16 weeks is possible. I have also told about pre-implantation genetic diagnosis, that before implanting the embryo, we can have pre-implantation genetic diagnosis. So particularly in 22q11.2, 50% chance of getting in each pregnancy. So each pregnancy has that chance. And at this, for this patient at 19 weeks, you can quickly do a FISH. Of course, you have estimated on 2D echo, so there is less doubt that it will come positive. But if you have to tell to patient, or if patient is not continuing the pregnancy, you can still take up the samples of the DNA and establish the diagnosis. If you have any other questions, Dr. Prashant.
is it possible to prognosticate? Because as you also rightly mentioned, and we know that DiGeorge syndrome can be a varying presentation,
yes,
right?
Yeah. So this mother also needs follow for hypocalcemia, hypoparathyroidism, autoimmune manifestations, and thyroid as usual. And if she takes a choice to give rise to birth of this baby, obviously for this baby, or if terminates, then for the next pregnancy, definitely a plan should be put on.
No, can we prognosticate as to where the patient will be, how the patient will behave clinically, or it is not yet possible?
No, yes. So if like TOF can be corrected, it is like a usual presentation of TOF. Hypothyroidism, hypocalcemia, very excellently managed. So prognosis is always good. Some of the patients may have learning disabilities, developmental milestones delayed. So every syndrome may not be a mental retardation. So in this particular case, if she is not having that phenotype, only cardiac manifestations, no other manifestations, and cardiac-wise she is stable, excellent prognosis. So not every syndrome has a bad prognosis. Excellent prognosis if you manage such cases, especially Noonan syndrome, Williams syndrome, and this syndrome. William has a higher possibility of developmental delay, but other syndromes may not have any mental problems sometimes. Many a times, actually.
Rajiv, come in the...
Yes. Actually,
your opinion and
yeah, Dr. Kane,
Dr. Kule or Dr. Praveen, some discussion on Praveen's case, whatever. Yeah.
No, not case. Actually, I just wanted to have my brain get washed, because I had a little primitive concept. I used to always believe that we go by phenotypic presentation and phenotypic diagnosis. Like in all our cases, we saw that everybody was presenting first the phenotypic. Even whatever cases I had of hyperlipidemia, I remember, even Nitin will remember, about 15 years ago in Asian Heart, there was one small girl with cardiomyopathy, supposed to be post-viral cardiomyopathy, and we demonstrated there was little turbulence in the left main. And then we saw that there were tendon xanthomas, and then Dr. Panda did the surgery with left main and right RCA ostial stenosis also. Even this Hasmukh Rawat's case, that 10-year-old with left main angioplasty also had tendon xanthoma. So we all go by phenotypic presentation first, and genetic testing is just added as an add-on, because so far we have that concept that genetic diagnosis is by case control association. We have 3 billion pairs, which we saw that 3 billion pairs with 3 million, the $3 billion worth research, and then genome total project is over. But then with so many pairs, there are some associations we see, and then with case control, we feel that they may be responsible. But then still nobody is very sure, and then for every disease there are some 15 possibilities and 20 possibilities. So to have a particular definitive diagnosis, a single nucleotide or single monogenic, as long as that is not there, to spend so much money and then to go for more extensive harsh therapeutic measures, are we justified? That is what I want to get clarified from the experts.
Yeah. So now, because we are highlighting, because as Madam said that many platforms are coalescing together, so directly whole exome sequencing, because cost has come down, especially during pandemics. The cost has very much come down. Whole exome sequencing may be 15 to 20,000. In cases where you are able to manage and help family members, it is much less cost. Earlier, it used to cost 30, 35,000, 25,000. Patient used to refuse. Now, patient themselves are asking how genetic test will influence or manage. So, in many cases, you have to prioritize depending on the familial needs and management. So that is first thing for genetic testing. Second is for Dr. Praveen Kulkarni's case, like you have a familial hypercholesterolemia. So we have agents coming up depending on the genetic diagnosis. Like we have some of the PCSK modifying agents, even if PCSK mutation is not there in such LDL receptor mutation patients. We have, I think, evolocumab, which is an agent which is in last trial. So these applications will be based henceforth on genetics. And as Dr. Burkule has presented a case of cardiomyopathy, in Duchenne muscular dystrophy, there are multiple mutations, and we don't apply directly NGS. We first do MLPA. I think Dr. Merchant, Dr. Rashid Merchant, a senior pediatrician, is in the audience, and he's always pushing me to have these discussions with specialists that in cardiomyopathy, We have in Duchenne's particularly, we have to first do MLPA. If it is not, if it comes negative, we have to do sequencing, which is nowadays some people are doing free, and then we have to do muscle biopsy. So almost very rarely in 1 or 2% cases, we are doing muscle biopsies. Like I have said, in amyloidosis, we are hardly doing biopsies. We are directly attempting genetic diagnosis. So in Dr. Kulkarni's case, I think genetic diagnosis would help in prioritizing drug therapy. And in the last case, skeletal myopathies, obviously there are muscle biopsy, very few cases is needed. We can directly apply genetic testing in most of these cases because the phenotype is so clear.
One thing I wanted to read
and
about
Duchenne muscular dystrophy. The Duchenne muscular dystrophy are mothers who are carriers are prone for cardiomyopathy. So I have few mothers. We had a big data which I have published with Dr. Merchant about these DMD patients, which we considered now whether for cardiac manifestations we need to do further cardiac MRIs, and as you have said, prescribing drugs. So those mothers have to be put on follow for cardiomyopathies. I think 20 to 30% of carriers are prone for mothers who are carriers of these children. They need to be monitored by cardiologists.
Yeah, I just wanted to read out because Madam Fatke left, but she has sent a message that exome sequencing is complementary to clinical phenotypes. So should be done in all cases with or without family history if non-genetic causes have been ruled out and are unlikely. It can be very useful for some patients and families. Even VOUS may present challenges. That's what is her conclusion. And I think she has stressed that again and again. So I think we have to reduce the cost, get the cost down, and maybe do. Yes, Millie.
Sir, I'm sharing one editorial. This is from the Indian Academy of Clinical Genetics, their journal. So this is the latest issue, and Madam Fatke herself has written this editorial.
And on Madam's
may I complete, Dr.
Just a minute.
Yeah. So I think what Dr. Rajiv Karnik was asking, when should we test and should we be doing these tests routinely? The last paragraph is especially instructive, what Madam has written. And I think that will answer our question. We may not pick up rare diseases in every condition, but there is a lot of overlap between some of the non-genetic and genetic things. Secondly, it will help us understand pathophysiology better. It will help us screening patients better. And now that we are getting targeted therapy for many genetic disorders, and we can start some of these therapies early in the disease, that will help. So we must now bear these things in mind. So I think this editorial is a must for all of us. It's a one-page thing. So please go through it. It's Madam's editorial.
Yeah. I think Uday, are you there? I have to ask, I'm so sorry. Dr. Uday? Dr. Uday Jadhav?
Only one question.
Yeah, please go ahead. I am there.
Yeah, for this patient who Dr. Praveen Kulkarni presented, the patient presenting within the first decade of life with such high cholesterol levels and with all the external features, is there any use of genetic testing in such a person? Because he's bound to be having familial hypercholesterolemia. Means he is kind of, if you go by that, why do genetic testing in such a patient, such a young person?
Yes, sir. So I have said that some of the, not particularly this patient, as you said, some of the therapies coming which will target PCSK variants if they help in such patients. Then as he has said that liver transplants, usually nowadays liver transplant patients are taken up if the diagnosis is confirmed, they don't take up otherwise. So genetic diagnosis is becoming routine and must for therapies like liver transplants. And third is family members. So many patients, like you said, that statins can be put up on surveillance and statins. So all these three factors have to be discussed with the family. That's if these three factors apply, genetic testing can be done a little early. But as Madam has said, because we have a rapid inflow of new drugs based on genetic testing, I think many patients of this sort will need genetic testing for confirmation and application of therapies.
Yeah, is Dr. Uday Jadhav? Uday, you are there?
Yeah, I'm here very much.
Sorry, yeah. Okay. Oh, you changed everything. Good. So I think of all the things, I have to take the burden because I think we have had a very good discussion. We almost have the same number of participants right from the beginning to now, and we have exceeded the time. That's only a negative point, but I think we have to take everything. So we have learned a lot. At least I have learned a lot, and I leave it to Uday now to summarize and propose a vote of thanks for everybody.
Thank you very much, Dr. Ghansham Kane, for coordinating the entire meeting. The protocol says that we need to thank all the faculties. I guess we are honored. I'm very happy for Dr. Snehal Malakmir for having taken this forward. It's a great opportunity for CSI members to learn something absolutely new than what has been discussed. The faculty members, our guest faculties and our faculties from Mumbai, without individually naming them, it has been a great learning curve for everyone. Here is what I have to say for the members so that they can take this forward. It's very important that now we start changing the way we learn things. I mean, the adaptation that whatever we have learned in the meeting so far is one thing. This particular program, I thought, has been one of the unique programs that we haven't had this opportunity to listen to aspects in cardiology which are very crucial for a set of given patients where, unfortunately, we are so much in the dark. Our understanding, even as cardiologists, is very limited. So I must really thank Dr. Snehal and the team who brought this forth, discussed with us, explained us the whole situation. And I will look forward, along with Medjonim. Thank you for hosting our meeting today as our unrestricted educational sponsor. But we would like you to get associated once again with us, where I will want our members to propose the cases where there are dilemmas in genetics, in cardiology, and I will invite them and request all our guest faculties today to help us understand those situations and answer those queries, so we can go the other way around. Let our members come up with their cases and let our experts solve it for them. This will be truly a carry forward learning curve, and I look forward to this. Thank you very much, Dr. Kane and the entire team for bringing this presentation to us today. Have a nice weekend.
Yeah, thank you.
Have a good day. Stay safe for the new year.
Thank you, Kane, sir. Yeah.
Thank you, sir. Thank you, everybody. Thank you, everybody. Thanks, Snehal.
Happy New Year to all. Yeah.
Dr. Shetty,
Thank you, everybody.
Thank you.
Have a good rest of the Sunday. Have a good 2 hours sleep.
Thank you.
Okay.
Okay, yeah.
Thank you.
Quite exhausting. We need to, each one itself probably is a meeting by itself. Because they have really good cases and we don't see them that often, and if we see them, we don't investigate them in that detail. So I think it was a great learning opportunity.
And it was a good topic to discuss today.
Yeah.
I think very rarely done. Very
New topic. Yeah.
Yeah.
Okay. Bye. See you.
Thank you. Bye.
No prizes
Bye.
today.
See you guys.
Uday sir, no prizes today.
Milind, yes, no prizes today. We can have prizes, of course, but how many times you're going to be a winner, Dr. Milind? You have to give opportunity. You have to give opportunity to others also to win.
To take something from Mumbai CSI. It's a good thing.
Let's work on something. Yes. So here is my suggestion. Let's take one more program forward. And let's have 3 prizes for the 3 best case presentations in genetics in cardiology. Anyone can do it. It's open to every forum, senior and junior. And let us have those experts in genetics judging us whether we are doing the right thing. Thank you. Look into it.